the forensic fraternity, the people from the judiciary, from the administration, from police departments, the UGs, the PGs, everybody. So we will be beginning very shortly with our sessions. So kindly wait for few more minutes. Our director sir will be joining us very soon, and then we will be able to start our inaugural and further proceedings of the session.
मैंने पे रिसर्च किया आज निकल इंटरनेशनल लेवल पे वो है तो इन्होंने मेरे को डाला था तो ज्वाइन कर लेना
ना वो सरस्वती बनना शुरू कराना
Please mute yourself.
I request all the delegates to kindly mute yourselves as we'll be beginning in a short while.
अब शुरू कर दें प्रेजेंट विंडो टैब से दो नंबर
नहीं अभी नहीं शुरू करना सरस्वती बनना
बस
अभी तो
लेना
बोल दो A very warm welcome to all of you once again. Extend my heartiest welcome to all of you for sparing your time. All of you have taken your time out for being here today. On behalf of the Department of Forensic Medicine and Toxicology of AIIMS Bathinda, I, Dr. Jyoti, Associate Professor, and the Organizing Secretary, welcome all the eminent guest speakers, the respected faculty members of the fraternity, distinguished delegates to this CME on DNA fingerprinting, past, present, and future. Thank you for joining us all on this academic event. I am pleased to inform that we have received an overwhelming response from all over the country. And the participants include distinguished people from different fields, renowned forensic and medico-legal experts, forensic scientists, forensic odontologists, people from the judiciary, law enforcement authorities, postgraduate students, junior residents, senior residents, medical officers, as well as MBBS students. Now, it is my privilege to address all of you together on this single platform. Together, we can accelerate the exchange of ideas and scale up all the existing good practices among us. Today's session comprises of talk by three eminent guest speakers, Dr. V.N. Mathur, Dr. K. Thangaraj, and Dr. Vimukti Chauhan, who are pioneers and have achieved excellence in their field. The faculty of AIIMS Bathinda will also share their experience in the sample collection in various states in which they have worked previously. Now, this will be followed by a panel discussion. All of you are requested to type the query or any clarification that are pertaining to the talk in the chat box side by side, and only a few questions will be addressed at that time. The rest of the questions will be taken up during the panel discussion. I hope these celebrations will be a good learning experience to all of us. As with all, for this and to become a success, we require blessings from God. We can formally
उधर से तेज करने
हे शारदे माँ हे शारदे माँ अज्ञानता से हमें तार दे माँ शारदे माँ, हे शारदे माँ, अज्ञानता से हमें तार दे माँ।
ये तो ये आपको स्टॉप हो गया नीचे से यहीं से स्टॉप कर दिया।
I now invite Dr. Akhilesh Pathak, Head of Department of Forensic Medicine and Toxicology and Organizing Chairperson, to explain the detail of the purpose of the CME.
Our director sir has joined us. A very warm welcome, sir.
Thank you.
A very good morning to one and all for joining us. Hope you all are doing well. On behalf of Forensic Medicine AIIMS Bathinda, I, Dr. Akhilesh Pathak, welcome you all to the online CME on DNA fingerprinting. Firstly, I would like to thank our eminent speakers for accepting my invitation and giving their valuable time to talk in this CME. Secondly, I would like to give my sincere thanks to all my dear colleagues and respected members for joining us today. As we all know that the theme of our today's CME is DNA fingerprinting, past, present, future. This is a topic that each one of us can relate with, particularly the forensic experts who are engaged with autopsy work. The aim of conducting this CME is to identify the preferred samples for DNA profiling and the correct methods of their collection, preservation, and transportation for further analysis in different circumstances like advanced cases of decomposition, skeletal remains, fetus, etc. During today's sessions, we will also try to find out reliable in the collection and preservation of DNA samples in forensic practice. It's a great pleasure for me to introduce our speakers who are going to talk to us about the DNA profiling and its medico-legal implications. Our speakers have spent almost their entire career in this area of DNA profiling. All these are award-winning professionals who have specialized in their fields and heading the institutes for over 15 to 20 years. Ladies and gentlemen, please join me in welcoming our today's speaker, ex-director Dr. V.M. Mathur sir, State Forensic Science Laboratory, Rajasthan. Dr. K. Thangaraj, who is Director of Center for DNA Fingerprinting and Diagnostics, CDFD, Hyderabad. It is an autonomous organization funded by the Ministry of Science and Technology, Government of India. And Dr. Bhimukti Chauhan, who is Assistant Director, Central Forensic Science Laboratory, Chandigarh in Punjab. And my colleagues from AIIMS Bathinda, Dr. Ajay and Dr. Jyoti will also share their experience of DNA sampling during practice of forensic medicine. Dear all, we have registered around 200 participants from different streams all over the India, including experts of forensic medicine and toxicology, experts of forensic science from dental college, experts of judiciary, experts from the police and law. We are having delegates from almost all premier institutes, including all AIIMS. Thank you all for joining us, including the PGIMER Chandigarh, all AIIMS, JIPMER, Manipal, GMCH Chandigarh, IGMC Shimla, KEM Mumbai, LP University, NICFS, National Forensic Science University Gandhinagar, and all these are making this CME truly a multi-specialty national event. Thank you all. This CME holds special value because this is the first CME which is taking place in collaboration with the Director of the Forensic Science Laboratory. We are proud to announce that the chief guest and patron of our institute and patron of this CME is our worthy director, sir, Dr. D.K. Singh, sir, who has already joined us, and he will give a small speech in few minutes. Now I would like to conclude my speech by encouraging the delegates to participate with an increasing number in all the talks and discussion through the digital platform for the next 3 hours. I wish everyone a successful, safe and fruitful event. Thank you.
Welcome, Director Sir. I request you to please say a few words and address the gathering.
Yeah, thank you, Dr. Akhilesh. I'm very happy that AIIMS Bathinda is organizing this very important CME. And I also welcome Professor Mathur, Professor Thangraj, Professor Dr. Jyoti Barwa, Dr. Vinumati Chauhan, who are participating as a faculty in this very important CME. The topic is DNA fingerprinting, past, present and future. I know the future is very, very important because whenever we as a clinician, we talk about the individualized medicine, individualized treatment, then there is the role comes the DNA fingerprinting or genome sequencing. I'm happy to inform you that I have worked under Professor Lalji Singh as his very close confidant when he was the Vice Chancellor of Banaras University, and he appointed me to develop the trauma center. That was the reason I used to sit and chat with him many times, and I used to listen his lectures many times. I have read his article in the Nature where he was having some hypothesis about the migration of Dravidians and Tamils and other things, and migration in India or migration whole the world. So it is also very important to know our DNA fingerprinting. Whenever he was sitting in the selection board, just to give an example, suppose if he is taking the interview of the anesthesia, I was telling every candidate that he is going to ask the genetic factor in anesthesia. Which drug affects which drug have a long duration effects and because of some genetic factors. He was very keen researcher, he was very keen learner, and he developed a stem cell building in Banaras University. It was really a very technical building because of his knowledge and his information. So you could build a very nice building. Even it was a learning for me because I was not knowing many things. What is the requirement of the bone marrow cell transplantation and stem cell researches. So that was the reason I used to discuss a lot with him. And I'm really proud that I have worked with him, that he is known as a father figure of fingerprinting in India. He started a genome foundation at his village and he started functioning from there only, and he has developed tie-up with the many universities, and really he wanted to develop this science, which is a very futuristic science. Because of his contact, I used to discuss many things like opening up the forensic universities in the whole the country, even in UP, and even developing some forensic science with the forensic medicine department for the better diagnosis and better treatment and better autopsies of the. So you know that we all know that our forensic autopsy is a very low level. We hardly conclude anything. So we just go on the by case scenario and develop the case. That is why this is a very futuristic department. I hope that all the discussion between you will lead to some recommendation, and Government of India is also thinking, because it is very costly department. That is why it is not very fast progressing, because it requires a specific equipment, a specific knowledge and a specific buildings. So that is why it is all important for you to recommend some reasonable way to how to develop this thing, to even maybe the forensic universities or even the Raksha Shakti universities, which is now 3 states is already started Raksha Shakti University. That is in their course, this topics is already included in their topics. So with all great hope, I wish you all the best and hope that some recommendation, something will come out of this CME, and that will be recommended to Government of India for future discussion. Thank you very much.
Thank you, sir. Thank you for your kind words. You have always been a source of motivation for all of us, and you've always encouraged the best out of among us. Now, beginning with our talk, I now invite our first guest speaker, Dr. V.N. Mathur. Dr. V.N. Mathur is ex-director of Forensic Science Laboratory, Jaipur, Rajasthan, retired in the year 2011. During his tenure as director, Dr. Mathur received 2 consequent extensions for his outstanding services. He set up the biology division in FSL Jaipur, Rajasthan in the year 1972 and is indeed one of the pioneers of DNA fingerprinting and serology division. He has delivered more than 25,000 forensic reports related to a variety of crimes such as murder, rape, exhumation, dacoity, paternity disputes, bestiality, and cow slaughtering cases. In fact, Dr. Mathur is among the first to deliver forensic reports of biology, serology, and DNA fingerprinting. He has attended various conferences of national and international importance and has also presented a number of scientific papers. He is the recipient of many prestigious awards, such as the Directorate of Forensic Sciences Meritorious Award by the Ministry of Home Affairs in the year 2010 and Dr. J.P. Modi Lifetime Achievement Award in 2012, which is one of the most prestigious awards. Over the years, Dr. V.N. Mathur has regularly been delivering lectures to police officers, judicial officers, administrative officers, and advocates. He is also a visiting professor of many universities, including the Rajasthan University, National Law University, Bar Association, Rajasthan Judicial Academy, Rajasthan Police Academy, Manipal University, and Amity University. Today, Sir will be speaking on the topic identification with and without DNA fingerprinting. I request Dr. V.N. Mathur to please share his screen. Sir, Mathur, sir, please share the screen.
Yes, I am sharing it. Yes. I trust my voice is audible to all of you. But,
Yes, sir, your voice is very much clear.
ಲ್ಯಾಪ್ಟಾಪ್ ಇಲ್ಲಮ್ಮ ಇದ್ರಲ್ಲಿ ಹಾಕೊಡ್ತೀನಿ ಬೇಕಾ ಬೇಡ ನೋಡು.
Okay. Before sharing my screen, I would pardon. Before sharing my screen, I want to speak a few words for the honorable guest. देखना अपने आप को इसके पहले बंद करना हाँ देखना आपके पहले yes before sharing the screen I would like to talk to everybody about this conference. First of all, I am thankful to Dr. D.K. Singh, Patron, Dr. Akhilesh Pathak, Dr. Ajay Kumar, Dr. Jyoti Bairwa, Dr. Ratan Singh, the members of the organizing committee for inviting me as an eminent speaker. I would also like to thank to my friends, medical jurists, including Dr. P.N. Mathur, Dr. Vyas, Dr. P.C. Vyas, Dr. Goswami, Dr. Shiv Kochar, and other also. Then I would like to yes. Are you? My voice is not audible to you? Sir, is it audible?
Yes, sir, you are audible.
Yes. All these, yes, all these medical jurists has supported me during my course of developing the DNA fingerprinting lab when I was struggling it. Establishing a DNA printing in lab in our Rajasthan forensic lab. I have dedicated almost more than 25 years to establish it. And I had been very closely associated with all these medical jurists. And we can't forget a few names before before starting this CME. The important names in the field of DNA profiling is Dr. Hargobind Khurana, Dr. Alex Jeffrey, the father of DNA fingerprinting, Dr. Lalji Singh, the pioneer of DNA fingerprinting in our country. I had close association with Dr. Alex Jeffrey, Professor Sir Alex Jeffrey, and Dr. Lalji Singh also. The boon of the DNA fingerprinting in our country was because of a very, very important report submitted by Dr. Lalji Singh. That was Naina Sani murder case. Naina, it was famous as a Tandoor case, Delhi Tandoor case. After that report, a revolutionary event occurred in our country for establishing, for training and DNA labs and all other things. So I'm thanks to for all these things and be associated with that. I also want to thank our special guests. Thanks to the organizing committee for inviting these three special guests, and I will tell you the significance of inviting them. Honorable Justice Mrs. Nandini Vyas, she is the director of State Judicial Academy, Jodhpur, and she is working hard and has taken the responsibility of producing best judicial officers. Nandini Vyas, Honorable Justice Nandini Vyas. So she has taken the responsibility of producing best judicial officers. Then Dr. J.M. Vyas need not require any introduction. He is Director General of the National Forensic Science University, Gandhinagar, Gujarat. This is probably the first university in Asia, or I trust probably in the world. It is of its own kind of university, and Dr. Vyas has taken the responsibility of producing best forensic scientists in the country and abroad. And finally and lastly, Professor Dr. Vijay Lakshmi Sharma. She is head of the department, School of Law, Manipal University. She has taken the responsibility of producing best advocates in the country. So you can understand, this is a wonderful triode who will certainly provide strength to the justice system. And in addition to all this, the medical jurists and doctors are a boon to this subject also and boon to this forensic profession also. I would also like to thank Dr. Thangarajan. Probably you may not be knowing about my relationship with Dr. Thangarajan. Dr. Thangarajan and Dr. Lalji Singh both had been my teachers and supervisors during my several trainings at CCMB for hands-on forensic DNA training. So you will surprised to know that Thangarajan is my teacher also, my guru also, and still today I have his manuscript notes with me, and I go all through them. So with all these things, I would like to share my screen and start our session. And during this session, I have selected some cases, past cases, DNA past, present, and future. I will show what was the past, what is the present, and what will be the future. And along with this, I will share some of my cases, both for paternity, rape cases, murder cases, and human slaughtering also. So I am going to share my screen. Thanks to all you. We will start our lecture with my PPT presentation. So thanks
I
to
think
all.
if you permit me to speak a line.
Yes.
Yes,
I
sir.
think this CME is a tribute to Professor Lalji Singh, as you have worked, and I have heard about Angraj. And I was lucky to work with him in another capacity, and used to sit with him or chat with him a lot. So it's this CME we can tribute to Professor Lalji Singh in his memory.
Definitely, I wish this. That is why I have given a reminder, because Lalji Singh is not with us. He was at Banaras University, BHU at Banaras. He invited me several times for the talks, but because of my busy schedules, I was not able to. But I was regularly going to
meeting
CCMB.
some Gopal Pandey.
I did not met him, Professor Dr. Tangarajan
Yeah.
and Dr. Mishra and were associated with me. Dr. Lalji was my very close friend, and he was sure that I will, and he helped me a lot. Where at one stage I got stuck when my 4.5 crore rupees was stuck with the central government STC, and the concerned secretary, he belongs to South. He tell me that you gave me some authenticity. You are guaranteed that you will be able to establish a DNA lab. So I just rang my telephone to Dr. Lalji and asked him, and Dr. Lalji told that secretary, "Oh, need not bother to the. I am with Dr. V.N. Mathur, and I will support him. I will suffice every facility to him." So I had great.
Even his contribution in DHU was great. He brought the
perhaps
stem cell
you
building and CDC, the Central Discovery, which was having 100 crore fund. He brought from ministry.
Yes, sir. And perhaps you might be knowing that Lalji is the first person who has developed BKM probe, who has developed a probe in our country for doing DNA fingerprinting cases. His probe is recognized as BKM probe, Banded Crit probe. So I gave show you one of the paternity case in which that Banded BKM probe of Dr. Lalji Singh has been reported. The case was reported by Dr. Lalji Singh, but because of his some problem, he could not appear in the Honorable Court of Rajasthan High Court, and he permitted me that I should explain his report. That was the first report on the Honorable Court, and that was the first DNA report in Rajasthan High Court, double branch. And probably you might be knowing it. There were two judges, Professor R.K. Saxena and Dr. Gyan Sudha Chandran. And then Gyan Sudha Chandran became the Honorable Justice in the Supreme Court also. So I had the privilege and proud to associate with so many big persons. So please give a token of respect and regard to Dr. Lalji Singh in his memory. I will share it also. Speak some words for Dr. Lalji Singh.
For Dr. Lalji Singh.
Thank you. Thank you.
Now you can share your screen, sir. Thank you, sir.
Sir, I have shared my screen, and it will be on PPT mode. It will be on PPT mode, and
sir, your screen is not visible to everyone.
Now is it visible?
No, sir.
No, sir.
Achha. No, sir.
Try to reshare it.
So please share it and put it on full screen.
I am resharing it.
Yes.
Ask to join. Hello.
बता देना क्या बता रहा पापा को जरा
The one said all that. Rajesh joins.
Now is it visible?
Yes, sir. It is visible.
Now it is visible, sir.
And my voice is also audible to you?
Yes, sir. You are loud and clear.
Without wasting the time, I am starting with the session. The session is DNA fingerprinting, past, present, and future. So this is my first slide showing what was the past status of the techniques in DNA fingerprinting. I have undergone a large number of technologies and trainings in DNA. In the initial stage, we were doing organic extraction of DNA by test tube methods, and then doing PCR, and then doing southern blotting. So you know that southern blotting is the technique of transferring the DNA from gel onto the high bond membrane, and we were labeling our probe with P32, that is radioactive isotope of phosphorus. It has a 15 days life cycle. So you can imagine how difficult and how risky it was to label a probe with a radioactive substance, but still we were learning and doing it at CCMB. Then VNTR, variable number of tender repeats, are to be studied, and all these are taken on a X-ray plate that is known as autorad, and it was a great risky to deal with the radioactive phosphorus or radioactive substances for labeling the probe. But in 1997, one more probe came into the market, and by Perkin Elmer, it was non-radioactive probe. It was chemiluminescent probe, which with the color changing probes. The DNA sample at a particular locus, it gets the change in color, and it was non-radioactive also. So it was very easy to handle it. At that time, that probe was mentioned as HLA-DQ alpha. It was a 6 specific locus marker kit for Perkin-Elmer. Elmer, and now in the present status, there are a lot of changes in the technology. We are having automated DNA DNA machine for automatic isolation of the DNA. Otherwise, it was very problematic to. It was very problematic to. It was very problematic to isolate the DNA specifically from the bones and the teeth. We have to cut the bone from the center and moist the bone marrow for several days, might be 3 days or 4 days. When the bone marrow gets some bit little softer, then we squeeze the cells from the bone marrow, and it starts doing. So in cases of exhumation, you will find bones also. But nowadays, we have automatic DNA isolation. The machine is known as Automate. And you will surprised to know and will be happy to know that while establishing a DNA lab in FHL Rajasthan, I established Automate. So today my scientist has no problem of extracting DNA from any of the sample. Just crush the piece of bone or piece of teeth, put it in the Automate. It will automatically isolate the DNA and submit it. Then we are having PCR. PCR was also there, but at the time, today we are also having RT-PCR. It has accelerated the work. At that time, we were reading the probes on paper strips or on autoref. Now you have a ready DNA scanner also. You put your isolated DNA in the scanner, and it will generate automatically printed reports. The report is known as EEG, electroencephalogram report. Any graph, any report which is related to the human body with the help of the electronic equipment is known as EEG. Then availability of multiple markers. While Lalji was doing work at CCMB in India, we were having only one. We were having. We were having. So we were having only one marker at them, BKM probe marker. BKM marker was developed by Dr. Lalji Singh. And now there are a large number of multiple markers are present. And nowadays, the color markers is also induced in the market. And advanced equipment technology also. This is the present status of the DNA technology in our country. In our country and abroad also. Now, this is one of photographs to say you that every scientist must take the regular DNA training, despite of his experience or his post or his cadre and other qualifications also. He must. So this is one of my memorable photograph when I was also taking DNA trainings in a high prestigious institute of the country, and now you can see. This is an antique thing, antique DNA fingerprint. It belongs to me, me only. I developed this DNA fingerprinting from my blood, from my saliva. The saliva was put on some stamp on the envelope, and the envelope was sealed with my saliva. So you are seeing this is the HLA.
Please unmute yourself, sir. Mathur sir, please unmute yourself.
Now I have unmuted, and the voice is audible.
Yes, sir, you are audible, sir.
Yeah. So
Share your screen again.
share screen.
A blank screen is visible.
I trust now it is visible.
Yes, sir. Yes, sir.
Yes. इसको hide कर ले. So going back to this, I will give you a quick watch that I had been many trainings, and this is my own DNA fingerprint, which I developed in the year 1997 on 25th January during my training courses for DNA fingerprinting. These strips which you are seeing, these are 3 strips, 2 batches of 3 strips. Each strip is an original strip of this HLA DQ polymarker kit. So at that time, we were reading our this DNA fingerprint profile as for this particular gene, I am heterozygous. For the LDLR, I am heterozygous. For GYP, I am heterozygous. But for SBG, hemoglobin binding gene, I am homozygous. For D7S8, I am homozygous. DNA7S is the locus. And for GC, general group of the genes, I am heterozygous. So this pattern was read at that time. Now this is an antique piece. Yeah, perhaps I know, I can understand. None of the persons attending this conference might be having this. And in the side, I have written past and future. Now you can understand this DNA fingerprinting has now become obsolete. It has become past. At that time, it was the best technique for DNA identification. But now it has passed. But it is speaking for future also. You can observe by photograph at the bottom of this DNA report. During my training, my teacher was Dr. Mohammad Tahir. He was a scientist from FBI USA, Indianapolis, USA. So when I show him this, got limited, and I show him, and when he came to my lab to visit my lab at Jaipur, he asked me, Mathur, why you have put your photograph here? What does it mean? I told him, sir, I trust that in near future, these the genetic engineering will produce photogenic DNA, and we will identify the person, the contributor of the sample, and we can generate a report like this, which I have generated earlier as 23 years back. So you can preserve this, and you can see it, my photograph on this. Now, what is the present scenario is that during the present scenario, we are having gene scanners, and now we are presenting the reports. You can see both the alleles in every column, 2 genes in every column. You please remember that we have taken birth as pairing of our parents. If somebody asks you what is the evidence, genes are also present in pairs. If somebody asks you what is the evidence that our birth has taken by pairing our parents, it is very easy to answer. It is very easy to answer. Observe your own body, right from head to leg. You have all pairs, all organs in pair. So you have pair of eyes, you have pair of ears, you have pair of nose, pair of hands, pair of lungs, pair of kidneys, and like that, and pair of lips. So this is an evidence that the genes are always in pair. Please do remember, it is a a specific thing for us. And now I am passing the future. Now this is the future DNA that I impose my photograph on my DNA profile. Now the future has come when translating the DNA to the appearance, and in future the scientists will put the photograph of the person who is not known earlier by the DNA fingerprinting. The future reconstruction will be done, and this is the future fate of the DNA. Now I'm sharing one thing. Uh, this is my handmade chart, and in this chart I have shown that medical jurists and forensic scientists, they are the two wheels of the cart for the assistance and to help the investigation of the justice system. The medical jurists conduct the medical examinations and the post-mortem examinations, and forward this, collect the samples, visceral samples, other samples for medical legal purposes, and forward them to the forensic lab. The scientists of the forensic lab, they conduct the examinations of the respective samples forwarded to them and produce the report and forward the report to the forwarding authority. Both these reports are submitted in the honorable court for investigation and finally in the honorable court. But sometimes some controversy develops as I'm sharing this thing. You see, in a double murder case, a prosecution was going on in a double branch of Rajasthan, Jodhpur High Court, Rajasthan. Double murder case, double branch. 2 dead bodies, 2 postmortem reports, and 2 FSL reports. In postmortem report, the cause of death was brain injury. The cranium was fractured, and cause of death was brain injury. But in forensic lab, the presence of potassium cyanide in the viscera. So there was a discussion which report is correct and what is the real cause of death. So the Honorable Justice N.N. Mathur of the High Court, he called me immediately. I was working as the additional director in forensic lab Jodhpur, Rajasthan. So I went to the High Court. He was sitting with his colleague, Honorable Justice, most probably Dr. Justice Tatia. And they were sitting in the room, and they put both these reports in front of me and told me, Dr. Mathur, you tell me which is the correct cause of death, brain injury or the potassium cyanide in the viscera. Then I told him that both the reports are correct. If you examine, you study both the reports simultaneously, it gives you the modus operandi of the crime also. Then they asked me what is the cause behind this explanation. Then I explained them. The modus operandi is like that. Both the victims, both the disease, they were initially given potassium cyanide with some sweet. They took the potassium cyanide and the cyanide started affecting. There is a general consensus in every person, and even it was in the Honorable Justice also, that as soon as you take potassium cyanide, it causes immediate death. But to all my friends, I want to tell you that potassium cyanide does not cause immediate effect. Every poison has got its own fatal time to cause harm to the body up to the death. In case of potassium cyanide, the fatal period is 3 to 4 minutes. So once they gave the potassium cyanide, the accused were also knowing that it will cause instantaneous death without anything. But when the time passes, more than 3 minutes, passes through the fatal period, above the fatal period of potassium cyanide, between this, they were restless. If both the patients will survive, they will identify us. So they gave heavy blow on the head of of both the persons. Because of this heavy blow on the head, the cranium was fractured and the brain was damaged. And the cause of death was brain injury, which causes instantaneous death, less than in friction of the seconds. But please understand, before doing this act of hitting, so that before doing this act of giving a hard blow to the head and brain injury was caused. The brain was damaged. Uh, Dr. Akhilesh, I'm getting some sounds in between. So kindly ask them to mute it. I'm getting sounds from many persons.
Mahavir ji, please mute yourself. Yes, sir. Sorry for inconvenience.
Because I will not able to explain. This is the best case to explain the coordination between the medical jurists, doctors, and the forensic scientists. So before the fatal period of the potassium cyanide, 3 minutes passes to 4 minutes. In between, both the accused thought that they have brought some wrong chemical, or it is a fake poison. So they gave hard hit on the head. This causes cranium fracture and brain injury, which causes instantaneous death. But please see, before during this 3 minute period, before heavy blow to the head, which caused the brain injury, the blood has circulated within 3 minutes. 72 beat, the heart circulates in the body at the rate of 72 beat per minute. So during this course, the blood has circulated minimum 3 minutes, that is 216 times. This potassium cyanide has been circulated in the body, in every organ, in every visceral organ. And as soon as it has caused its fatal time, that is the 4th minute, in which it would have caused death to both the disease, the blood has circulated 72 into 4, 288 times. That is why before getting a blow or before getting the cranium fractured and brain injured, this potassium cyanide in the body has circulated a minimum 216 or 280 times. That is why the potassium cyanide is present in the viscera, and the real cause of death is brain injury. So both the Honorable Justice in the Rajasthan High Court, Jodhpur, they were satisfied with it. And when they inquired, the truth was revealed. They asked me where the potassium cyanide is generally available. I thought it is available with Sunars, gold maker, gold smith, all that. And the mystery was resolved. So this is a very good example of a coordination between the medical jurists and the forensic scientists also. So I will share, this is the diagram I made on the paper on this table, and asked him that one beat, heartbeat cause one systole, one diastole, and one pause. And during this session, whatever the photographs I am telling, these are all cases reported by me, and has got nothing to do except for learning purposes. Then identification of individual is a very great problem. If you identify the victim, then many problems of investigation are resolved. But you see, there are 3 means of identification. If it is visible or not. Hello, Akhilesh. Hello, Akhilesh. Akhilesh.
Sir, it is visible.
Visible, na? It is visible on the
yes,
screen.
Sir, screen is visible. Yes, sir.
But not to the participants. Some of the participants. Anyway, so you do it. So identification is being done by 3 methods. One is fingerprint. These characteristics are have been embossed by the nature on our fingertips. So from fingerprint, identification can be done with 100% match or 100% mismatch. Then DNA profiling is also the technique. The DNA is preserved by the nature in every nucleus of the cell. Every nucleus of the cell. So for this, we have to break the cell and the nucleus, and it also gives 100% match or mismatch. But in between, superimposition technique. Superimposition can be done from the skull. The skull can be used for sex determination, for age determination, for facial reconstruction, and the superimposition is done by meeting anatomical landmarks on the skull. But please remember, with superimposition, we cannot give a report for 100% match or mismatch. It is only by fingerprint, which are morphologically visible on our fingertips, we can give 100%, and from DNA also we can give 100%. But from, if you have to give a superimposition report from this, you can write there are strong possibility of match or mismatch. So I am now showing a case for related to paternity and all that. Now there is a big question, which is the topic of our conference also. Why DNA? Why DNA is required? So the answer is that DNA is the same material which constitutes the genes and the chromosomes. And the genes and the chromosomes are responsible for sex determination, species identification, hereditary characteristics, transmission of hereditary characteristics.
Excuse me, sir. Excuse me. Sir, slides are not visible, sir. Slides are not visible. Please share again.
Acha, we are
Please share the slides again.
Yes. Now we are sharing it again. I don't know why it is.
Yes, sir. Somebody else tried to share. That's why he's disconnected. So please share it. Yes, sir. So these are the problems in online. We all know.
No problem. Except that our lecture will be extended a little more for
Yes,
2 more
sir.
minutes.
Now it is visible, sir. You can go ahead,
Now
sir.
it is visible. Yes. Yeah. So today's CME and conference is on DNA. DNA present, past, and future. Then my question is, why DNA? The answer is that DNA is the only person, only sample which is available in almost every person, every cell, except red blood corpuscles, which has no nucleus. It is present in every nucleated cell, and DNA is the same material. It is a genetic material. It is the same material which constitutes the genes and the chromosomes, which are responsible for sex determination, amylogenin gene, and specific species identification, then transmission of hereditary characteristics, and individual specific characteristics of the person also. That is why we concentrate on DNA profiling, the today's best developed technique. Now, the answer for this, that if you have a blood group A, B, and ABO, in case of blood group, you can give a negative report with certainty that if the father and mother both belongs to O group, they cannot produce a child with A, B, or AB or O group. They will produce only with the O group. But if the father has A group and mother B group, vice versa, the phenotypes are A and B. And if their genotypic combination is, the combination will be A or AO or BB or BO. If a couple with the genotype combination A and O, B and O, then they can produce. Akhilesh, I'm listening many voices in between. So can you request them to mute their mics? So if the genetic, the couple with A, O, and BO genotype, then possibility of producing or the child from all the blood groups A, B, and ABO. So in case of blood examination, report from blood grouping, you are confident to give a negative report. I am showing it in this paternity cases. You see, this is a case reported by me. A pradhan of some village was alleged for sexual assault with a girl, and which conceived and was pregnant. The girl was Pradhanji's neighbor and a good friend, but she alleged Pradhanji, and later on she delivered a female child. In between, police officers contacted me what to do. Then I told when we will resolve the mystery after the birth of the child. Now you see the official report. The blood group of the Pradhanji is O group, and the blood group of the mother who was alleging the Pradhanji was also O group. But the blood group of the child was B group. So on the Mendel's principle of heredity, Pradhanji cannot be the father of this baby child. Therefore, Pradhanji is not biological father of the child. In case of blood grouping, a negative report for elimination or for elimination can be given with certainty. Please do remember, it is for elimination, not for including. But possibility it cannot be given if the couple belongs to A or B group. These are my reported cases. You see, this is one of the case where the girl was missing from a fair, and she was kidnapped by some peoples, and later on she recovered, and it was sent to me for paternity identification. In this case, father, the baby in question, Raveen, she is in question, is O positive, mother is O positive, father is O positive. The mark of identification, you see the mark of identifications are scars and moles. Scars may or may not be present on the body because they are the repair marks of the injuries, but the moles are birthmarks. So on the basis of the presence of mole, the identification mark, then blood group of the child, blood group of the parent, then I conclude that this could be the possibility of this father and mother could not be ruled out for being the biological parent of the child, and this child was handed over to these parents by the honorable court. Please do observe, baby, Raveen, father, mother, they all belong to O group. Now, in a similar case, and no DNA was taken into task for this. No DNA report was taken. But in the next case, you see, a girl was kidnapped from Karachi, Delhi at the age of 3 years. Then she was sold to some group of prostitutes. And during the police raid in brothel in Rajasthan Talk, this baby girl, who was named as Rajni, it was recovered along with two other girls also, and the photographs were published in many of the newspapers. Then her father, on the basis of mark of identification, his father Meher Chand and mother Bhagwani Devi, they came to claim it. This is the baby Rajni when she was sent to me at the age of 7 years. You will surprised to know that this girl was recovered after 10 years. When the parents on the screen, Meher Chand and Bhagwani Devi, with all O group, when they went to to the court, honorable court, the court refused to accept this FHL report, and they told this report was given to me when this girl was 7 year age, and the court handed over this girl to some rescue centers for the girls. You will surprised to know that this girl was again kidnapped, and the peoples were and the parents were never gave attention by any officer, by officers or political persons. So on one day, these two parents, they were sitting in the Jaipur High Court, and this mother Bhagwani, during the lunch hour, this mother Bhagwani Devi was crying like anything, weeping and crying. So group of advocates came and asked the cause and purpose. Then they show my report also and told that we have lost our girl after getting it once at the age of 7 years. Then the advocate was confident. He had been my student in any of the session. He told to the parent, this report has been given by Dr. V.N. Mathur, and there is no state of argument that this girl doesn't belong to you. Then he filed an habeas corpus, and Honorable Justice gave the order to produce this girl in the court. Then that sub-inspector, who was officer Mr. Dashrath Singh, who was sub-inspector then, at that time sub-inspector, now he became the additional director. So in compliance of the Honorable Justice, Honorable High Court, he has to get this, recover this from the same brothel area. But now in this case, you know, the blood group of the mother has already been disclosed in our forensic report. It is O positive. So the people from that community, brothel community, they raise another mother to whom this girl was identifying, because this girl was living with them, calling somebody auntie and some mommy and chacha and all that. So she was isolated from the group because she was having O positive. Then she also started claiming that this is my own girl. I am keeping it since so many years, and she is my daughter. And all stories were made. Then Honorable Justice of the High Court, R.K. Saxena, he called me, Mathur, what to do it? All are O positive. Then I told them, now we will go for DNA profiling. And this was the first DNA report. It was prepared by Dr. Lalji Singh at CCMB. But I was asked by Lalji Singh to present in the Honorable High Court, and the High Court permitted me to present this report. And you can see the status of the report at that time also. So this was the first historic case in Rajasthan High Court of the double bench, R.K. Saxena and Justice Gyan Sudha Chandran. Both are Honorable Justice, and Gyan Sudha Chandran was had been the Justice of the Supreme Court also. So DNA profiling was done for them. The blood samples were collected in judicial custody, in Honorable Court also. With the other. Now this is the report. At that time, such type of DNA fingerprinting reports were developed. This is report prepared by Dr. Lalji Singh with his BKM probe. And you can very well see, I prepared this report as to illustrate this case in the Honorable High Court. It took me about more than 2 hours to satisfy both the courts, both the prosecution and the defense, and the defense had to commit that this girl really belongs to these parents. You can see the red color. This is ladder. It is 10 kb ladder. This is kilo base ladder, where we studied our base pairs of the DNA. And this position, this is the position effect of the DNA. This position is ascertained to every individual specific by the nature. You cannot alter this thing. So the same BKM probe was used by Lalji Singh and asked me to present it. Now you can very well see, this is the, now it is a primitive report nowadays. It is a very important past report of the analysis. You can see in between, these are 6 samples. The ladder, after ladder, there are 5 samples. One belongs to father Meher Chand, then the Rajni, baby Rajni, mother Bhavani Devi, mother first, mother second, she was a prostitute Mona, and that fifth is the my own blood. I was not, it is unrelated person. It is unrelated. I am not related with this case. So I am also O positive. So you can see all are O positive. Now see the DNA bands, the 2 alleles. DNA is always in pair. I told you already also. This one band of baby Rajni matches with the band of mother Bhagwani Devi, and the other band matches with the father Meher Chand. But none of the band matches with this second mother who was claiming, mother Mona, the prostitute Mona, and V.N. Mathur. So you can very well say, you can convince the Honorable Court that one of the gene has been inherited from the mother and other has been inherited from the father. So these are the biological parents of this child. So in this paternity case, I was bound to use DNA technology, and this was the report that Meher Chand and Bhagwani Devi are the biological parents of Kumari Rajni, and Srimati Mona is not biologically related to Kumari Rajni. So the girl was handed over to the legitimate parents, and you can see the photographs with me and my staff also, along with the girl and the parents. And this is a comprehensive photograph for in between at the age of 7 and then second mother to inclusive and the final. And this news are published in so many newspapers of the country that after 11 years, 11-year girl restored to the rightful parents, girl returned after home, and I produce a paper that I prevention of a girl from prostitution. But nowadays again, in paternity, there is a problem. When some male or female child born at the same time in the labor room, the mother starts claiming that she has delivered a male child and other family. So this is a case when there are 2 mothers and there are 2 child. One of them is female and other female. You can very well see in the last column. This is amylogenin gene. Column 1, mother first, column second, profile of the first child below, photograph below this mother, then photograph of the child in between, and lastly the mother second. You can say here we are doing profile for mother only identification. So you can see that one of the gene of this child, female, is belonging to this mother one on the right hand, and other belongs to the male child belongs to the mother. Okay. This is same case of paternity also. The person received his sister-in-law, and she delivered a child, and he refused to do. And nowadays we are preparing and presenting the DNA report like this. And for the between the middle column shows that the child is we need not require any body to be examined. Simply blood will tell you the sex. So you can find that she is a female. On the right hand, red color profile of the father, blue color profile of the mother. You can see one gene has been contributed from each father and mother. So it is a decided case by paternity. Now, the next is, here I have not used DNA fingerprinting. It is a very, very interesting case that one person, he is a great horoscoper. You see, he is a grandfather. He read horoscope. He is a great horoscoper, very renowned horoscoper of our state. And he has predicted so many predictions about the politicians and other persons. And his forecast never got failed in future. So on the day of the reception day of the marriage of his son, he declared that I will be blessed with a grandson on date so and so at time so and so. He specified the date and the time also. And you will surprised to know, on the very same day, from the labor room, he and his family got the news that you have been blessed with a son. And it was entered in the hospital register, delivery of the male child. Please, it was delivered. It was mentioned in the hospital register. A male child has been given birth by this lady. After a period of 2 hours, almost 2 hours, they were handed over. Were handed over with a female girl, and the authorities in the hospital, they made a cross on male child and put it replaced it with a female child. Now this register became an authenticity of the replacement of the child, whether it was male or it was female. So he rang to many of the senior officers, police officers, and other politicians and all that. I got a message. I forgot to tell you about the report of this Rajni girl. You will surprised to know that initial examination of this girl, medical examination of this girl Rajni, was done by Dr. Shiv Kochar at AIIMS Medical College. And he asked the police, and he forwarded this case to me. And as a close association of Dr. Shiv Kochar, between me and Dr. Shiv Kochar, we were able to hand over the right girl to that rightful parent, biological parents. And in this case, they got the news, and it was entered first male child, and later it was replaced by female child. So there was a. So I was asked by my senior police officer and DG and others to immediately rush to the hospital. And when I went to the hospital, I came to know that during this suspected hours, 2 hour span of 2 hours, 6 couples have been blessed by the child. So you can very well understand that the jugglery can be made among the 6 childs only. Otherwise, the child might have been handed over it. So the complainant, the grandfather, they lodged a complaint in the police and specifically mentioned that they will not accept any report other than DNA fingerprinting. You see, it was specifically mentioned in the FIR. They will not accept any report. I may give any report, but they will not accept any report other than DNA profiling. And my director gave a press statement that my expert will do without DNA report. So I was shocked how this confession can be given at this hour when we have not examined it. But during the, so I kept waiting all these 6 couples, and you will see these are the 6 childs. Yes. So how to find out? So the question was that during when I was examining this case, this grandfather, he came to me to meet me in the office. He was a great astrologer also and face reader also. And lot of people. He was a highly respected person, retired principal of some Sanskrit college. So lot of people from my office also surrounding him, going feet for him. Then he talked to me for 2-3 minutes and told me that now I will accept any report which you will give. Why you have came here, sir? He came that I have to see the person, the expert who will be giving report, important report for my family. During this talk, I came to observe a morphological characters. All morphological characters are given are the product of the genes, whether they may be dominant or they may be recessive also. But several morphological characters are present on our body also. So I observed a squint in his eye. You can see a squint in his eye very well. So I immediately asked him, yes, sir, you have sustained any eye injury in future, in past? He told, no, I never sustained any injury to my eyes.
Sorry to interrupt you, sir. We are already running short of time. So can you please finish it early?
So with this case, we can finish it early, and I will show. So this case was solved by the study of the squint. You can see the squint from the grandfather has been transferred to the father and the child. And in my report, I told him that it is on Bendel's principle, and you are the father of this child. So I will skip the rape cases and come to the human slaughtering. I will just take 5 minutes only to show. In one case, I did not take assistance of DNA, and in other case, I would like to show. This is a very important case. You might have not come across this person on the screen. He committed 86 murder, and on the 86th murder, he was chasing a girl, and the dupatta of the girl create obstacle, covered his face and head, and he thrown out the hair, the dupatta. That dupatta was carrying some hair samples, and that doesn't belong to the female. They were sent to my lab for examination, and I was astonished to see that this is a multi-melted human hair. It is one in billions and billions. So I told the investigating officer that search for the suspects and send me the hair samples. They sent the hair sample to me of this person also. It was multi-melted hair. But there was a problem that on hair we can't give a death sentence to me. So I requested that I will go for neutron activation analysis. At that time, DNA was not there. So I sent the samples to BARC, Baba Atomic Research Center, and on the basis of neutron analysis, both the samples were matched 100%, and the person was sentenced to death. And finally, in case human slaughtering case, a girl was slaughtered, decapitated, and the body was slaughtered in 3-4 pieces, and these body pieces were recovered, and the head was hidden by the culprits. After an investigation of 2-3 days, the person told that he will be able to recover the head. He will give you the recovery of the head. So in late nights, I went with the police officer, and just in some garden, just near a plant, by digging the soil right from his finger and nails, he caught hold of the hairs and pulled out the entire face. You can see the last photograph. You might have seen this thing in horror pictures or horror movies also. Then after watching the face, because the face was not decayed, it matched anatomically, and it's matched with blood samples also. So DNA fingerprinting was, does not conduct it, and the girl was identified as Sapna. It is Sapna murder case. But in other case in Jaipur, so a body was slaughtered into 19 pieces after decapitation, and these pieces were disposed of. and they were some slump area and all that, and they were recovered. And you see that these are the pieces, uh, multiple pieces. You see 18 pieces, and it took me to recover with the police assistance and other assistance. All the 18 pieces were recovered. You see. Then I was asked by the police headquarters that whether one person has been slaughtered or number of people's massacre has been done in that area. So after 3-4 days study, uh, I gave them a report, and you can see the last photograph. Here the person was very intelligent. He removed the penis also, the male organ also, to give a fake impression that the body belongs to female. But I could see that remnant portion of the testing, so I was confident. Then I took all the pieces, washed them, get them anatomically meshed, and reconstructed the body. You can see the reconstructed body in center and the center. And then I gave my report. I took a blood group of all 18 pieces also, and blood sample for DNA profiling of all this, because I was knowing that it can be a question in the past, in the future, for specific identification. You can see the slaughtered pieces, how sharpness, you can observe the sharpness on the pieces. And the head was missing, and incidentally, the head was also recovered later on. And his parents came to from Mirror to search him. So I got a message that the parents are they might be the parent of this boy also. So we took the blood of them, and blood of all these 18 pieces, and got DNA profiling, and the DNA profiling matches with all the pieces and the parents, and the boy was identified as Khalil murder case. So these are the two pieces of human slaughtering. So this is, I already told you that the future of the DNA is facial reconstruction, phenotypic DNA is also. Reports are being generated in American cases. This is one of the reported case where the facial reconstruction has been predicted earlier, and the person was arrested. And this is future. And my dream project is that in future, no child should born with the deformity, because of the reason that the entire human genome project has been completed. We know each location and effect of each and every gene. We know the position effect of there are chemical seasons activators also, and advanced technology. So there are some of my suggestions that in future we expect that a photograph will be put on the DNA report of the contributor who has contributed for the sample. And it is my suggestion that every medical college must have a DNA lab or genetic laboratory for their routine work and for their research purposes also. And regular training courses for DNA fingerprinting should be conducted for doctors, associates, and medical staffs, and the associate forensic scientists also. And now finally, I am telling just in a span of 1 minute, 60 seconds, a DNA pathy. You might be listening a new term, DNA pathy. What is it? This is Alex Jeffrey, father of DNA profiling. And I was chatting with him with my later pen friend. He came to in 1994, a world conference was in Hyderabad. When I met him for the first time, he identified me that perhaps you are Dr. Mathur. I told him why. He told me that it is DNA pathy. So the DNA pathy, this is Dr. Larji Singh. This is Mr. Southern, who taught me, he himself taught me doing Southern blotting for DNA samples. And privileged by this is Bruce Buroli. At that time, he was the chief DNA scientist. And this is one of the Nobel laureate to whom I had been deeply associated, Dr. C.J. Gurjasic, Nobel laureate. He was able to detect the gene for perforation of brain in sheep and got rid of it. Dr. Muhammad Tais is one of the best DNA scientists of USA, honored by status of DNA doctor. And this is the time I got the meritorious award for forensic scientist, Home Minister Meritorious Award in the year 2008. And finally, I was thankful to the doctors who rewarded me with Sir Dr. J.P. Modi Lifetime Achievement Award. after my retirement in February, in February 2012. So with all this, I want to leave the dais that I am a proud first trainee of the, not only of the NSS, but the first training of the CCMB for these benefits. And thanks to all of you. So I am thanks for listening me patiently. So
Thank you, sir. Thank you for enlightening us about the use of DNA profiling in a variety of cases. Your cases indeed were very interesting, in which it is very impressive to know how the cases of maternity-paternity disputes, in exchange of baby and murder, all of these were solved by means of reports, of the serological reports, DNA profiling, as well as by phenotypic methods. And your suggestions that were given at the end, definitely we'll try to incorporate all of them. Your presence, sir, will be needed in the panel discussion. Your expertise will be of great help. Sir, please join. Sir, we
thanks
have...
to everybody for listening me patiently.
Yes, sir, we have a question from Dr.
Bharatuday. Sir, there is one question from Dr. Bharatuday. Sir, do we have DNA database facility available in our country for medical legal purpose?
No, at present we don't have DNA data facilities, and I really wish that it should be developed. Perhaps you might be knowing a DNA data bill has been already produced in Lok Sabha also, and it will be going to the Raj Sabha for collection also, until the government doesn't allow to collect the DNA data in some specific cases. Might be in some disease cases for the curing of the disease and for medical legal purposes also, unless until at present we don't have any data collection to the best of my knowledge in our country.
Thank you, sir. Thank you.
Thank you, sir. Please join us again in the panel discussion. I now invite our second guest speaker, Dr. K. Thangaraj, Director of Center for DNA, Fingerprinting and Diagnostics, Hyderabad. The Center for DNA Fingerprinting and Diagnostics is an autonomous organization funded by the Department of Biotechnology, Ministry of Science and Technology, Government of India. This center is equipped with world-class state-of-the-art instrumentation and computing infrastructure to facilitate research and development in frontier areas of modern biology. Dr. Thangaraj. Prior to this, Dr. Thangaraj was the Chief Scientist and Group Leader at the Center for Cellular and Molecular Biology, Hyderabad. His main research interests are origin of modern human, cardiovascular diseases, mitochondrial disorders, and male infertility. He has published over 250 research articles. Dr. Thangaraj is an elected fellow of Indian Academy of Sciences, Indian National Science Academy, and National Academy of Sciences. He is a recipient of J.C. Bose Fellowship, Sun Pharma Research Award in Medical Sciences, Raman Research Fellowship, Lifetime Achievement Award, Excellence in Science Award, Distinguished Scientist Award, Dr. Subhash Mukherjee Oration Award, Professor Manjeet Singh Oration Award, Dr. Palle Ramarao Award, and many others. He is a board director of Mitochondrion, associate editor of BMC Genetics, Tropical Medicine, International Health, and member of the editorial board of various journals. He is the founder of the Society for Mitochondrial Research and Medicine. Today, Sir will be speaking on the topic DNA fingerprinting in forensic and its medical legal application. Dr. Thangaraj, I request you to please join us on the platform and share your screen.
Yeah, thank you very much for the introduction. I also thank Dr. Akhilesh for inviting me to give a talk. Nice to see Dr. Mathur and Dr. Singh, Director of AIIMS. How long I have time to speak?
Yes, sir. Sir, 45 minutes.
45. Okay, thank you. My slide is visible?
Yes, sir, your slide is visible.
Okay, very good.
Sir, please put on full screen mode.
Yeah, I kept on full screen. You're not seeing full screen?
No, sir.
No,
No,
sir.
sir, please press the preview button at the right lower end corner.
No, that I could see here. I escape now and again sharing it. Could you see full screen or not yet?
No,
No,
sir,
sir.
it's still not visible in full screen mode.
Okay.
Please share it again, sir.
Okay. Could you see anything?
Yes, sir. Your presentation is visible.
For me, it's not visible.
Okay.
Is it full screen?
No, sir.
Okay. Fine now?
Yes, sir. It is visible now.
Full screen?
No, sir, it is not in full screen. But we can continue it, sir. We can continue. It is still visible, sir. Yes, sir. We can go ahead, sir.
Even now, full screen not visible? Full
No,
screen?
sir. Full screen is not visible, sir. The entire screen is visible.
Okay, you could see the slide without.
Yes, sir. Yes, sir. We can see the slide.
Okay,
Yeah.
then I'll go ahead with this.
Yes, sir.
Yeah,
Thank you.
right. So, since the title of the CME is DNA fingerprinting, past, present, and future, I thought I'll tell you some of the historical perspective of DNA fingerprinting. Then I'll take you through the forensic and medical applications of DNA fingerprinting. How the name came into existence, why it's called DNA fingerprinting. So the name came, as all of you are aware, the fingerprint is very unique to every individual. In fact, that was found more than 160 years back by Sir William James Herschel. He was a chief magistrate of Hooghly district in Jangipur. He used very effectively the fingerprint on the native contractor. Therefore they don't cheat after taking money. What was interesting is that the observation which you made is not a scientist. The observation which made that the fingerprint is very unique to every individual. He and some others have pursued with that idea, and after 30 years, so they published that information in a journal. It's a very highly cited journal called Nature. That was in the year 1888. So that also tells that any observation should not be neglected, particularly in the context of the applications of that particular observation. Coming to DNA fingerprinting, as Dr. Mathur already mentioned, that Sir Alec Jeffrey was the one who was working on a beta globin gene, and suddenly found that this is called as autoradiograph. When he hybridized a particular piece of DNA onto different individuals' DNA, he found that every individual's profile was very unique. So that was the first time he identified. Subsequently, in the same year, he identified and also proposed different applications for forensic and immigration and so on. So that was the first time it came into existence in 1985. But during the same time, because this is a kind of repetitive sequence, our human genome consists of repetitive sequence all over the genome. Dr. Lalji Singh, about whom Dr. Singh, Director AIIMS mentioned, and also Dr. Mathur mentioned, he was the one who was working on the evolution of snake sex chromosomes. In human, what we have XY, the Y chromosome is the one which has a gene which makes individual as a male. But in case of snake, there's a WZ chromosomes, and in the primitive snake, we cannot differentiate the chromosomes. In the intermediate, we cannot differentiate, but we can differentiate at the DNA level. That was the snake, the banded kite, that consists of a class of repeat called GATA, what you see here, the red color, GATA, tandemly repeated. Then he got an idea that why don't we try with this particular probe and see whether this also generates individual specific DNA profiling. He tried that, and in fact, this is the very first case which was given evidence.
Excuse me, sir. Sorry to interrupt you, sir. The slides are not moving. It is still stuck on the first slide.
It's not moving. Yeah, then there's a problem.
Yes, sir. Yeah. Now it's moving, sir.
Okay, now it's moving. So I kept the initially as the full slide mode, but is it moving now?
Yes, sir.
Yes, it's moving.
Right. Yeah. So this is the one which I was mentioning, the Lalji Singh carrying the snake, and that snake DNA, particularly the banded kite, consists of class of repeat called GATA, which was mentioned in red color. So that repeat is used efficiently and shown that it could generate individual specific DNA profile. And this was a very first case where he went personally to Kerala, a place called Telacheri, and gave evidence that he is the father of this child, who actually promised that he will marry this lady, but he said later on refused that the child is not belongs to him. But the DNA profiling nailed down that he is the father of the child. Let us look at little more about the DNA. You all know that every cell except RBC consists of the nucleus, and the nucleus consists of the chromosomes. In our somatic cell, there will be 46 chromosomes. 23 we inherit from father, 23 from the mother. In addition to this nuclear genome, which we inherit equally from both the parents, there are other DNA, particularly DNA which is present in an organelle called mitochondria. You know that this is the powerhouse of the cell. It consists of its own DNA called mitochondrial DNA. While we inherit the nuclear genome or nuclear DNA equally from both the parents, we inherit mitochondrial DNA or mitochondria only from the mother. There's no maternal contribution of mitochondria or mitochondrial DNA. This property is used to trace the maternal lineage. Again, the nuclear genome, we have the Y chromosome specifically present on the in the male, which can be used for tracing the paternal lineage, which I'll come a bit later. This also is very important to understand that how the genome is inherited. In the somatic cell, I mentioned that in female, there are 46 chromosomes, 44 chromosomes, that is 22 pairs, are common between male and female. There is no difference. So only difference is the sex chromosomes. In case of female, there are 2 X chromosomes. In case of male, 1 X and 1 Y chromosome. I mentioned that this is essential for making individual as a male. But in case of the germ cells, we have only half of the chromosomes. When there's a meiotic cell division takes place, the 46 chromosome become 23. So from each pair, it receives only 1 chromosome. But the segregation pattern at every cell division is not programmed in such a way that okay, this chromosome from this pair has to go, or from this chromosome from this pair has to go. So there's a combination. We never know that in what combination it goes. For example, out of these 2, so 1 chromosome goes here. Out of these 2, 1 chromosome goes there. But in the other egg, instead of this chromosome may go. Right. Same way in case of female, male, again the sperm will consist of only half of the chromosomes. I mentioned that the segregation is not always programmed; it's random. That's the reason every individual is very unique. Although we inherit the genome from our parents, except in case of the monozygotic twins, where they have identical genome. But when these 2 fertilize, then the individual or the fetus or the child will receive the 23 from mother in the form of egg, 23 from the father in the form of sperm, so they become 46. So this is the basis. So every individual inherit 50% of the genome from the father, 50% from the mother. I also wanted to tell you some of the facts which is essential for DNA fingerprinting. The DNA is very stable, unlike other molecules. Like RNA, if you take, is not stable, or any other molecule you take, they are not very stable. But DNA, it's very, very stable. It withstand very high cold, also withstand very hot condition. Once you heat the DNA, it become single strand. But if you allow that to cool, it become double strand. DNA is double strand, as all of you are aware. And the DNA can be isolated from any biological material. So it can be a blood cell, particularly WBCs. It can be isolated from semen, saliva, urine, hair, but hair root, not the entire hair, tooth, again tooth pulp, bone, any kind of tissue. So the biological material from any individual can be, the DNA can be isolated. So not only the source, but also can see that without our own knowledge, or anybody who makes a crime can leave their DNA material. For example, people are now having a habit of closing the envelope with the saliva because there's a gum already. So that can have few cells. Chewing gum, when you take coffee or tea, the rim of the cup can have few cells. Similarly, smoking the cigarette butt can have a few epithelial cells. That is sufficient for generating the profile. And the DNA doesn't change with the age. If you take few cells from just born baby and make a profile and wait for 50 or 60 years, again take this blood and generate the profile, it will be identical, except in case of some tissue where there could be some rearrangement, particularly in case of cancer, there could be some rearrangement. Otherwise, it will be constant. Pattern of an individual from every tissue, if you take, the profile will be identical. For example, saliva, blood, or any tissue, root, hair root, if you make a profile, it will be identical. And most importantly, it shows variation between the individual when we use appropriate markers. Either can be a southern-based marker, PCR-based method, or if you do the whole genome sequencing, because there is a variation between the individuals. You can identify every individual with 100% confidence. So where this variation exists, if you look at the chromosomes, there could be variation at chromosome level. Most of you must have studied that there's a trisomy. Instead of 46 chromosomes, there's one chromosome extra. In case of chromosome 21, it's called as Down syndrome. Chromosome 13, Patau syndrome. Chromosome 18, Edwards syndrome. That's a trisomy. There is also monosomy like Turner syndrome. There will be only one X chromosome. But if you go into at molecular level, there could be a lot more rearrangements, deletion, duplication, and so on. But most importantly for DNA fingerprinting, you can look at the DNA sequence. DNA is made up of 4 nucleotides, as all of you are aware: adenine, guanine, thymine, and cytosine. There could be one base substitution. Instead of C, there's a T, or instead of A, there's a G. Okay, so that's one level of variation at single nucleotide level. So there could be variation at repeat number level, like I mentioned about GATA. One person in a given position can have 10 repeat, other person can have 15 repeat. In the same person, one chromosome may have 12 repeat, other person may have 15 repeat. Okay, so that's the level of variation which is useful for DNA fingerprinting. When the DNA fingerprinting started, the technique which was used was southern-based method. So one has to use a restriction enzyme to cut the DNA. For example, here, every restriction enzyme has a specific restriction motif. In case of EcoR1, wherever it finds GAATTC, it will cut the DNA in that particular position. For example, you are using a probe. Let's take GAT as a probe, and that probe finds its complementary sequence here. There is a, it will anneal with T. There's a C, it will anneal with G. Okay. In case of GATA, CTAT, that's what it will anneal. So, if, for example, this is a region, the purple color one is the place where GATA goes and binds and gives us the profile. In two individuals, for example, the same position, but the difference is instead of A, there's a G in another individual. So if you compare the profile of these two individuals in a given position, the restriction enzyme will cut the DNA here. Therefore, what you see, there's a 2 different size fragment, whereas in another individual, the restriction enzyme will not cut to this position because the A become G, therefore this site has been altered. So instead of 2 small fragments, there will be 1 big fragment in this individual. So that's the basis for southern hybridization method. So entirely depending upon restriction digestion. So for example, just giving you example, the one individual, what you see is a big band. There's no restriction site, what I mentioned earlier. But so there's one restriction site in this individual. Therefore, this band is not present. Whereas instead of that, there are 2 bands here. Okay. So this is the basis, but this is the entire process of southern-based method or RFLP-based method. It has a huge process. At least it would take 1 week to 10 days to get the profile. Those days, you can imagine that it requires more DNA for southern hybridization. It also requires more time, and it requires radio labeling for the probe for detection. So the process goes like this. You take a DNA, isolate the DNA, and use the restriction enzyme. There are several enzymes which are used. EcoR1 was the one which I mentioned earlier. H3 and many more are there. It will cut at a specific nucleotide position. Then we have to do the gel electrophoresis, and the DNA is negatively charged. Make the agarose gel, load the DNA here, and apply electrical power DC. The DNA moves from negative to positive here, and during that time you will not be able to see any band. It will make a smear because Its DNA is very, very long, and all the DNA fragments are organized one after the other, because so many fragments are there. Like a book, if you take most of your medical books, they are very huge in volume, but there are large number of papers. So you see as a bulk. That's how you will see the profile, not a profile, actually the DNA, the electrophoresis, after electrophoresis, you will see after staining with ethium bromide, you will see as a smear. Then transfer that onto nylon membrane, because DNA, now it is in the gel. Gel is not stable, because once you heat up, it will melt again. So therefore, you cannot handle it further. You transfer that onto the nylon membrane. During the process, you also make the DNA into single strand. DNA is a double strand. The process of transferring the DNA onto nylon membrane, you make it that as a single strand. Then use a probe in case, for example, GAT or any repeat, and label that with the radioactive material. Then you iodize onto the nylon membrane where the DNA has been transferred. After that, there's a process of iodization, washing, everything takes one full day. Then you expose that onto X-ray film and leave it for 1 week in minus 70 degrees centigrade. Then take it out, develop like a regular film. After that, you will see bands like this. That means these are the places where the repetitive sequence have been hybridized, because those are the ones which are labeled with radioactive material. And then you read that and interpret. So this is the past method. Nobody used this method now. But now the present methods are most simpler, PCR based method. You can get that the profile within a few hours. That is entirely based on the number of repetitive sequence present in the DNA. So these are the common repetitive sequence are used for DNA profiling, otherwise called as STR, short tandem repeats, or simple sequence repeat, or microsatellite repeats. You can see here CTAT repeated for many times. Here CAG repeated for many times. Here CTAT repeated for many times. So what the present method does is that, so these are the repeats, what you see in one allele, one of the chromosome. And here you design a primer flanking to this region. Then you label the primer with radioactive material, not radioactive material, the fluorescent tag. Then you amplify. And naturally, you will get smaller band because the number of repeat here compared to this is smaller. And here you will get little longer band. You get like this, smaller band and the longer band. And if you are using large number of such microsatellite markers, you will get large more bands. So how does it looks? I also want to introduce the pedigree here. You must have studied in the genetics about the pedigree. When you put square, forget about the color, put the square, it indicates it's a male. And if you put circle, it indicates female. And if you put line between these two, that indicates there's a marriage between them, and these are their offspring. Now I introduce the color, where you see that there are 4 colors. Here there are 3 colors. Just to give you idea about how the genome is inherited and what are the genomes. Here in the male, this indicates autosomes, right? There are 22 chromosomes here and 22 chromosomes here. Then in case of male, there could be X chromosome. You just leave that X chromosome aside. And this you consider as a Y chromosome. And the center one you consider as a mitochondrial DNA. Here again, half of the chromosome material, and this is half of the chromosome material, 23, 23. Then this is the mitochondrial DNA. Now look at their offspring. The male always receive the Y chromosome as such from the father, and it passed on generation after the generation in the male lineage. Whereas the autosomes, if you look at, one child, this off has come from the father and this off has come from the mother. In this child, this off has come from father and this off comes from the mother. Same way here, this off and this off. So you can see the color of autosome, each one is different. But look at all of them, either son or daughter, all of them receive the mitochondrial DNA from the mother. So this is a basis for coming slides to explain. So this is how the genome is passed on generation after generation. Keeping this in mind, these are the one which is presently used, autosomal STR, and of course the markers are same, but the method which has been used several years ago using different kind of automated DNA sequencer with the gel based gives beautiful image like this. You can easily, anybody can now tell whether this is the father, mother and the child, and one can look at every locus. I mentioned that every single locus there are repetitive sequence. Take that, this particular locus. There are 2 alleles here, there are 2 alleles here, 2 alleles here. Father, mother and the child. Now what do you expect? That half of the genome comes from the father, half of the genome comes from the mother. So you see that there are 2 alleles. One allele is matching with the father, other allele matching with the mother. So this indicates the number of repeats present there. The more number of repeat, the migration of this particular sample comes slow. So whichever is there on the top, that means that has more allele compared to the lower. So it's very clear in this particular locus. Take again here. So one allele is matching with the father, other allele is matching with the mother. So like that, all of them are, if it's accounted 50% from father, 50% from mother, then you can clearly say that yes, they are the biological parents of this particular child, or he is the father of the child, or she is the mother of the child. On the other hand, if you look at the same locus which we are talking about here, there are 2 alleles here, 2 alleles here, 2 alleles here. So now you can see the one allele in the child is matching with the mother here, and this allele, if he is the father, it should be either here or should be here. But since it's present here, so this one allele you can rule out. Then same way here, one allele is matching with the mother, other allele is not matching with the father, either this or that. Same way there are many loci which are been shown with arrow, clearly demonstrate that the mother is the mother because one of the alleles present in the child is matching with the mother, but other half is not matching with the father. Therefore, mother is the mother, father is not the father of the child. So this is the way. Then how it is used in the, can you people mute your mic? Then how it is used in case of forensic applications. Let's take this, just giving a few examples. So this is one of the rare case where you see the victim's DNA isolated from the blood, and this is a vaginal swab, and this is the male who involved in the crime. Now you look at carefully. There are 2 alleles which is present in the blood of the victim. Here there are additional alleles, right? Because the DNA is present in the vaginal swab is very less. Therefore, that particular allele is very, very low, but it is matching with the suspect. What you see here, 2 alleles. One allele is matching with here, another allele is matching here. Same way, you can actually see that all of them are accounted for this particular individual is involved in the crime, suggesting that yes, he is the culprit. Further to confirm, I'll tell you about Y chromosome which later, but you can see the Y chromosome profile, unlike the 2 alleles in the autosome, because we have pair of chromosomes. There's only one Y chromosome. You can see that the profile is what you see is exactly matching with the culprit's profile, clearly confirming that yes, he is the person who involved them. In case of medical application, this is one example one can take it. So there is a lady diagnosed with the breast cancer, and there was a sample taken from the cancer region, and it was processed for the pathological analysis. After that, it has been confirmed that she has the cancer. But later on, the lady refused to accept and thinks that there could be an exchange of sample that may not be her real sample. Therefore, she wanted to confirm whether the report was correct or not. So what they approached was that taking the fresh sample, blood sample from a wound, and compare that with the paraffin embedded sample. So paraffin embedded sample, you know that it has to undergo a lot of process, but still we could be able to isolate DNA and compare that. You can look at here. This is a profile generated from the paraffin embedded tissue, and this is a profile generated from the fresh blood sample. But since, as I mentioned, that the tissue which has undergone a lot of process has highly degraded DNA when the DNA is of little higher size. The smaller size, no problem, it has amplified nicely, but you can see that the amplification slows down when the fragment size is more, but that doesn't matter for us. Now you look at the profile of profile generated from the tissue and the profile generated from the DNA isolate from the blood. Both are absolutely matching, confirming that tissue where they have identified a cancer cell or cancer-like morphology is actually our own sample, not anybody else, confirming that is a very important thing. Then I also wanted to tell you about the Y chromosome and mitochondrial DNA. So Y chromosome, I mentioned that it is present only in the male, only one Y chromosome, and it has a very unique feature. As it has a gene called SRY, that's the sex determining region on the Y chromosome, present on the P arm, this is the short arm of the Y chromosome. There are several other genes responsible for spermatogenesis, required only for the male, maleness and male characteristic features. I also mentioned that it goes generation after generation, the male lineage. So now the same principle which I mentioned, so I will not go into detail, but look at here. This is the mother, child, daughter, and the father's profile. But if you look at the son and the father's profile, it's always identical because the Y chromosome goes as such from father to son to grandsons, right? That's sometimes useful for some of the complex cases where there's a mixture of DNA samples. So this is the autosomal profile. There's a lot of mixture of sample. Very difficult to identify how many individuals are present here. But it's very easy here because of many reasons. One particular reason is that there's only one allele. You can avoid all other complications, even the female DNA is present. So in this case, what you can clearly see, so all these, the sample obtained at a different source belongs to one individual, right? And there are two more individuals are involved in that crime. So it's very clear when we use Y chromosome markers. I also wanted to highlight some of the very interesting case how the Y chromosomes have been used to trace the paternity of one of the Sally Hemings' son. To tell you a little bit about that, Thomas Jefferson, he was the third US president. He has a habit of having slaves. One of his slave, Sally Hemings, she had several children. One of her child, Easton, he was the last son of Sally Heming. He resembled Jefferson, and that was several generations back. But few years back, maybe about 8 to 10 years back, 3 groups, Chris Taylor Smith from Oxford, Mark Jabling was Alec Jeffrey's colleague, and Peter Denny from Leiden. All of them involved to try to understand whether the Easton is the real son or the Jefferson is the real father of Easton. So they did the Y chromosome profiling using different set of markers. This is a biallelic marker, microsatellite markers from Y chromosome, and this is a mini satellite markers. Sally Heming's son, Easton, and each one is 1 generation. You can see they're trying to trace after 5 generations here. Same way here, 5 to 6 generation. The way they looked at is, Thomas Jefferson had only 1 son. He died in the car accident. He tried to use his paternal uncle's lineage, because I mentioned that Y chromosome in all the males will be identical. So they used, you can see every individual, that number belongs to 1 generation. And these are some of the suspects. But these 2 markers, biallelic markers and mini satellite markers, are not very useful. But the microsatellite markers, which gives much more variation, and that matches the Sally Hemings and Easton's profile, matches with Jefferson male lineage profile, except 1 mutation here, confirms that yes, Thomas Jefferson is the biological father of Easton. You can imagine after several generations back, they have traced that. I'll also tell you some of the examples in coming slides. Now, I also wanted to give you some idea about the mitochondrial DNA, very, very useful when the autosomal marker fails. The Y chromosome and the mitochondrial DNA gives much more valuable information. So this is one example where somebody has been killed and thrown in a well, and the body was very badly mutilated, and they recovered and tried to do the profile, and they informed there are 5 families claiming that the deceased person belongs to that family. Mainly because the government gives compensation to this family. So this is one example where we analyzed 1 family. The mother is not there in this family. 2 brothers and a father. This is the autosomal STR profile, but unfortunately it was not giving profile because the DNA was very badly degraded. So you can see only up to here, less than 200 base pair. Beyond that, it's not given. So we thought that we should use the mitochondrial DNA, because mitochondrial DNA advantage is it's very small genome, 16.5 kb, and it's circular. And there will be many copies against 2 copies of nuclear genome. In a given cell, there will be hundreds of mitochondrial DNA. Therefore, it's easy for analyze and get the information. But again, you can amplify them into small fragment and construct a longer sequence. So with this minimal number of allele, difficult to predict whether this person belongs to this family or not. Now we used, there are 2 brothers, father's mitochondrial DNA is not relevant, therefore we did not use. We used both the brothers' profile, and you can clearly see that this is one of the brothers' profile, mitochondrial DNA profile. This is another mitochondrial DNA profile, another brother's mitochondrial DNA profile. Now you see this is called as hypervariable region. So wherever there are dots, those are the place there's a difference between these 2, or you can say that these 3 profile. Now if you look at each one separately, this is a real brother, this is also a real brother, this is a profile of the deceased person. You can see there's a T in case of the real brother, and also T in case of another real brother. But what you see here is a C. After G, there's a C here. Okay, there's one mutation. Similarly, here there's a CA in the real brother, CA in the real brother, but what you see here is CC. Okay, so there's another mutation. There's one more mutation here. There's A, also A in the real brother, but what you see here is G. It's a small stretch of sequence, but there is a lot more mutation. But we see difference, but clearly we can say that the diseased person is not belongs to this family. So that's the power of the mitochondrial DNA. This has been very, very effectively used, particularly for organ transplantation, where somebody like the donor and recipient, if they are not immediate related like father, son, or mother, son, or daughter, if they are maternally related 2 generations back, or they are paternally related, one can use the mitochondrial DNA and the Y chromosome marker. So somebody, if they claim that they are maternally related, we use mitochondrial DNA of donor and the recipient, you will not see any difference. The sequence is identical. But sometimes people also say that they are maternally related, but you can clearly say that again, this arrow mark shows there's a mutation between these two, there's a difference between these two, therefore they cannot be maternally related individuals. So that's again is useful for medical legal application. This is again very, very unique case where somebody has been killed and thrown in a some place, unknown place. Again, one family or multiple family was claiming whether that that deceased person belongs to their family. In this case, this is the skeleton which was sent to us to analyze the DNA and generate the profile. This is a mother's profile and this is a father's profile. As all of you are aware by now that there's no contribution of father of the mitochondrial DNA to the son, and you don't see his contribution here. Whereas both the deceased person and the mother's profile is matching, T and T, whereas here C, again A and A, there's a deletion here, GG, TT, and these two are different in the father. So this again clearly demonstrates that the deceased person belongs to this family. In a large context, it's not the DNA fingerprinting, not only that to address the immediate relationship, but it's also very useful for understanding our own population history. So this is one of our old study where we have looked at various population by now, and you know that, I don't know, some of you may not even know that in the Andaman and Nicobar Islands, there are population like this, typically look like Africans, and so their origin was very, very highly discussed, but earlier people could not come up with any kind of conclusive evidence for this population. So we sequenced the complete mitochondrial DNA, that was the year 2005. And we identified a lot more mutations which was not present anywhere in the world. You may not see because it's a very small screen. Each number represents the mutation in that particular in the mitochondrial DNA of those individuals. And the beauty is that one can also calculate the age of the population based on the mutation rate. And using that, we came to the conclusion that the Andaman tribes are the first modern human who migrated out of Africa, taking southern coastal route and reached Andaman and Nicobar Islands about 65,000 years back. You can see the power of the genetics and power of the mitochondrial DNA. So it's very, very informative for tracing the origin of the people. This you might have studied using a similar technique, using mitochondrial DNA. They trace that Prince William's mother, that's a Princess Diana, has a South Asian or Indian ancestry. And somebody during the British period, her 444 mother actually gone from India to Europe, and ultimately people able to trace this the history. And what is whatever I've been telling was the present day analysis. So what is the present and also the futuristic type of analysis? One is the genome-wide analysis. So one of the study, I think, which Dr. Singh also mentioned about the nature study which we did, where we used large number of population groups using 1 million single nucleotide polymorphic markers that came on the Nature cover, where we used large number of Indian groups. This is European HapMap sample, African HapMap sample, and the Chinese HapMap sample. So this itself give you clear indication that every continent or the country, people are very unique, leaving the Indians. In the Indian group, every population is very unique because we follow endogamy marriage practice. So that leave aside because that's not the topic of interest right now. But you see that people from Europe and Africa and China, they're all very unique. But if you identify some genetic material, it can be either drop of blood or air root or anything, when you don't have any idea, particularly when we have crime caused by neighbor countries, taking that sample, you can do the profile using 1 million SNP, and you have now large number of data set. This is called as principal component analysis. Just put the data there and it will take you wherever that person belongs to. So it will go and align there. So that's one which is being used now, apart from what we used for tracing or identifying the Indian history, genetic history, particularly population history. That's one of the important things. And what is another important thing which I wanted to mention to you is the ancient DNA based study. So where we are talking about the fresh samples. But when you have sample of 1,000-year-old, how to get the information about the particular sample? I'll give you again a couple of examples. You know, most of you, that the place called Rupund, that's about 5 kilometers above the sea level. There are a large number of bones have been found there by initially Anthropological Survey of India. And we took the study, we collected some bone sample, isolated the DNA, and try to understand who are these people. There are a large number of theories have been told. People say that they are the trader who went there, and there was some natural disaster. Everybody has fallen into the lake and perished. And people also they say that people have gone as a pilgrimage. Again, something happened. Everybody perished there. And people also think that there's a king who went along with this family and all the soldiers. Something happened. They all perished. So many more stories. But what we found, who are they? And when we looked at, there are 3 distinct group of people. So one, because I could not zoom it further. So one group looks like they are typically Indians. Others says they are Mediterranean people who probably migrated. I don't know why they have gone there. Nobody knows that. Only history can tell. But genetically, they are totally different. They are Mediterranean. Other group is Southeast Asian group. So that is, all these are possible by looking at the whole genome of the ancient DNA technology. This is another study. People, you also must have recently studied about a couple of years back with a large number of groups, including various archaeologists and anthropologists. We studied a complete bone sample obtained from Rakhigiry, and that shows that it has 2 different components. One of the component is the Andamanese component. They are the one who first come to Indian subcontinent. Other one is Iranian hunter-gatherers. You can see the color proportion tells that this is a combination of both. But what was surprising at the time was that there was no Iranian farmers component there, because there was a thought that the Iranian farmers are the one who brought the agriculture into India. There's a place called Fertile Crescent. That's where the agriculture and the domestication of animals all started, but we didn't find that. giving a clue that probably Indian must have started agriculture on their own. The agriculture must have come from Iran much later. So these are some of the other aspect which I wanted to tell you. But now people also use the phenotypic information like the eye color, hair color, and we also thought that there are certain mutation specific to India. For example, the 25 base pair deletion which we mentioned, exclusive present only in South Asian groups, which is not present elsewhere. That's one of the phenotypic features. So all of them together, one can also make use of as an informative markers. Also, people use the other biological means like the bacterium which grows in an individual, or the disease causing pathogens present in every individual also can be used as a marker for identifying the individual. So these are some of the futuristic things which will be useful for forensic analysis. So with that, I stop, and if you have any question, I'll be happy to answer.
Thank you, sir. Thank you for such a wonderful talk on the medico-legal application of DNA fingerprinting. It was really interesting to know the historical perspective related to DNA profiling and the use of Y chromosome in tracing the paternity. Your case discussions were really impressive, particularly the role of mitochondrial DNA and as well as the analysis of Asian DNA that has been useful in identification. Thank you, sir. Your expertise will be required during the panel discussion. We have a few questions that will be addressed during the panel discussion. I would like to ask one question which is from Dr. Ajay. How do you rule out stray DNA or contaminated DNA from the test samples?
It's very easy. Again, somebody's mic should be muted. Very easy. We know that every locus, so we all have only 2 alleles, right? If you find more than 2 alleles, then you expect that there's a additional DNA mixed with that. So it's very simple.
Thank you, sir.
Thank you, sir. So please join us during the panel discussion. Next,
Sure.
I would like to invite Dr. Ajay, additional professor in the Department of Forensic Medicine at AIIMS Bathinda. Sir has guided more than 8 MD students and have authored more than 54 scientific articles in the leading journals. He has 3 chapters in various books. A lot of innovations have also been done by Dr. Ajay, such as the Microsoft Word, the template that the people of Punjab and Haryana are using for filling the medical legal report, that is the MedLeaper. This was devised by sir. A marming machine was also devised using a simple DC motor and other peripherals in a very less amount, very economically embalming machine was being devised. He was also instrumental in developing the safe kit for the one-stop crisis center. Sir will be speaking and sharing his experience during the tenure of collection in the various DNA samples that has been done during the autopsy practices. Sir, please, I request you to share your slide.
Present now. Window. स्क्रीन कर लो ऐसा। हाँ, इसको मिनिमाइज कर भी कर पीपीटी ऑन
शेयर करो, शेयर
नहीं पहले पीपीटी ऑन करो, मिनिमाइज करो पीपीटी ऑन है। हाँ, प्रेजेंट स्क्रीन कर दो।
इस पे क्लिक करो।
नहीं होता यार।
क्लिक करो होता है। क्लिक करो, अब शेयर करो।
हाँ। हाँ हुआ है।
Is the screen visible to all of you? I hope it is visible.
No, sir. No, sir.
Please share again.
Okay.
See, someone else's screen is coming in between. Raju Ram Manju's screen is coming.
Yes, it is visible.
So don't minimize that window which is to be shared. Don't click minimization on that PowerPoint window.
Yes, now visible.
Okay. Good afternoon to all of you. Good afternoon to all the respected guest speakers, the senior faculty, the junior faculty, and all the distinguished participants. I am Dr. Ajay Kumar, additional professor working in Department of Forensic Medicine, AIIMS Bathinda, and I will be telling or sharing my experience of current practices of DNA sampling in Punjab and Haryana. So because of paucity of time, I will be rushing through my slides. So basis of my slide will be basis of my slides, basis of my current practices in Punjab and Haryana are the guidelines and protocols which are to be followed, and they were given by Ministry of Health and Family Welfare. Then the second guidelines or basis were by the Government of Punjab Medical Legal Manual, which has to be followed in Punjab and Chandigarh, and it is Manual of Instructions on Conduct of Medical Legal Examinations, including Postmortem Examination. Now I will be telling about collecting DNA samples. How do you collect? So in cases of, can be broadly divided into two categories where you are collecting samples in cases of living persons or in cases of dead. Again, in cases of living persons, two categories again can be divided, like in cases of sexual assaults and then rest of the medical legal cases. So I will be covering first sexual assault cases, sample collection. Basically, I won't be going into the nitty-gritties of the examination, consent, and rest of the sample collection, but will be telling mainly on how to collect DNA samples, how to collect samples which are required for DNA analysis. So first is you have to ask the patient, ask the survivor to stand on large sheet of paper so that all the trace evidence will get collected onto the large sheet of paper which is placed beneath the survivor. So it can contain grass, mud, pubic or scalp hairs, etc. So this sheet has to be folded carefully and preserved in a paper bag to be sent to FSL for trace evidence detection, including DNA fingerprinting. Then you can preserve the clothes that the survivor was wearing at the time of incidents of sexual violence. Now it depends on how much time has elapsed between the person, between the incidents and the person who is reporting the incidents. So you have to decide accordingly. If it is a fresh case, within 3-4 days, you have to preserve the clothes, and there is a proper format or proper way of preserving the clothes. You have to look for presence of stains, any semen, blood, foreign material, etc., which should be properly noted. And you always ensure that the clothes and samples are air dried, not sun dried or anything else, but they have to be left overnight and they are air dried before storing them in their respective packets. Usually the packet should be breathable ones, like which allow the air to pass through them, like paper bags. You don't store them in plastic bags. Now, fold clothing in proper manner so that DNA which is present at one side or the sample which is present at one side of the cloth does not get transferred onto the other side. And of course, you have to pack each piece of clothing in a separate bag, seal it and label it duly, and then send it for onward transmission. Then in cases of living persons, the best and easiest way to collect for DNA sampling is venous blood, which is collected and transferred to 3 sterile vials or vacutainers. First vial or vacutainer is for blood grouping and drug estimation. Second vacutainer, which contains preservative sodium fluoride, it is for alcohol estimation. And the third one, that is the EDTA vial. In that you have to collect 3 to 5 ml of venous blood for DNA analysis. Now someone may ask why you are sending the survivor's own blood for DNA analysis. Actually, it will act as a control and a sample which can be compared with that of the accused if the accused is caught and subjected to medical examination. Or if the accused is caught and the DNA estimation, DNA examination is requested by the police, then we can cross-match with that of the survivor. Then another is hairs with roots can be sent for DNA examination. Then 2 swabs each from the vulva, vagina, anal opening for anogenital evidence in cases of if there is any history of sexual violence. Swabs must be collected depending on the history and examination, but again ensure that they are air dried. Do not pack them or seal them as fresh because they can be contaminated with fungus or they can undergo degradation and decomposition. Now, the important points regarding dispatch of samples is while handling cases of samples, a requisition letter addressed to the FSL stating what all samples are being sent and what each sample needs to be tested for is to be clearly mentioned for scientists of FSL so that they get an idea what is to be done with the sample. So for example, if you are sending vaginal swabs, then you have to clearly mention over the envelope or the packet that vaginal swab is to be tested for semen or for spermatozoa or for DNA analysis, so that the people in the forensic FSL department, they will know why the sample has been sent and what is to be done with the sample. Now this form must be signed by examining doctor as well as the officer to whom the evidence is handed over. Then sample collection in rest of the medical legal cases, living cases, we collect, the best is we collect the blood sample. We have to collect the blood sample, though we can send samples of hairs but with roots, so easiest is to collect blood sample in EDTA vial. Now I will share about sample collection in dead bodies. So in fresh bodies, blood in EDTA vial again is to be collected, or blood on FTA card can be sent for DNA estimation. So this FTA card is available actually in the casualty department. So in medical legal cases, you can collect 1 or 2 drops of blood on an FTA card, let it air dry, then properly seal the card, and then you can send it for FSL department. Similar is the case with fresh bodies. You can send the blood in EDTA vial, or if FTA card is available, then it is best to send it on FTA card. Otherwise, in some places, you can send blood on gauze piece, because always the police official says that he does not have the FTA card, and so it is requested that the blood on gauze may be preserved. So you can still preserve the blood on gauze piece. Then in cases of decomposed bodies, blood if available in EDTA again. Then highly decomposed bodies, if there is muscle mass, then you can preserve a piece of muscle mass in saturated salt solution, or you can preserve the bone or tooth or hair. But make sure that the bone or the tooth does not contain soft tissues in them, because that will start degrading and putrefying. So it will be difficult for the FSL people to extract the bone once degradation starts. Then in cases of skeletonized bodies, again, muscle mass, if it is present, then bone or tooth. If you have bundle of bones, prefer sending smaller bones because transportation of long bones, femur, it is very cumbersome and difficult. Though in some centers, sternum or mandible with teeth, etc., they were regularly sent for DNA analysis. Then in cases of fetuses, blood, if available, again in EDTA vial or on FDA card, and then cleaned femur bone with no preservative is to be sent directly to FSL. So these were about all the samples which can be collected in for DNA analysis. So these were the current practices of DNA sampling. Thank you.
Thank you, Dr. Ajay. We will be taking up the discussion during the panel discussion later on. Moving on, now I invite Professor Akhilesh Pathak, Head of Department of Forensic Medicine and Toxicology. He will also share his experience of DNA sample collection. Sir was awarded the MD Forensic Medicine by SMS Jaipur and PG Diploma in Health and Law by Academy of Hospital Administration, New Delhi. He is having experience of about 20 years in teaching and has conducted around 3,000 autopsies. He has delivered about 20 lectures as a guest speaker in various scientific conferences all over India and has published 55 research papers and written textbooks of forensic medicine. He is also editor-in-chief of an international journal, an editorial board member and peer reviewer in 12 journals of specialty from India and abroad. He is also a guide of MD and PhD students in forensic medicine. Presently, Sir is working as chairman of the recruitment cell and procurement cell in AIIMS Bathinda. He is also a coordinator of the medical education unit and member of the ethics committee in AIIMS Bathinda. He has conducted orientation program for medical students in January 2021 and organized the first hybrid CME conducted here at AIIMS Bathinda in February 2021. The slide has already been shared, so now I request Sir to please share his experience of collection of the samples.
So good afternoon all. So here, without wasting the time, I'm moving on toward the slide. And whatever the experience we have during the practice of forensic medicine, what kind of samples we were preserving. So I worked in Gujarat and we were dealing mostly with the autopsies. And there in Gujarat we were not having anti-mortem work. So I will focus on the samples which were collected during the autopsy. So when we were receiving the dead bodies in fresh condition, see, in most of the centers, we were preserving the sample of blood on a gauze piece, and it was dried in air and preserved in a glass bottle, and then it was handed over to the concerning police person for the DNA profiling. So that was the most common method we were using to preserve the blood sample for DNA profiling. I've also observed that at a few places, the medical officers were preserving sternum in a glass bottle because we are removing the sternum during the routine autopsy, and that sternum was preserved in a glass bottle and sent to the forensic science laboratory for the DNA profile. So that another sample was the preferred one at some of the centers. It is also commonly used method for the DNA profiling in some of the centers at Rajasthan. In decomposed body, so we have done a lot of autopsies during my tenure in, especially in Rajkot. So we were having the lot of autopsies of the decomposed bodies, and there we were preserving the sample of blood again on gauze piece. So it was a hemolyzed blood, but we got the positive results with that. So, and but in cases of the advanced decomposition, we were preserving the molar tooth. So molar tooth was preserved in a glass wire bottle for the DNA profiling. When we were receiving the skeletonized remains, we preserve again the molar tooth if it is available. It was the first choice. And in few cases where the skull or mandible were not available, we preserve the ribs or vertebra or these other smaller bones, and we got the positive results with that. We have also conducted so many cases of the chaired bodies. In all such kind of the cases, we preserve the molar tooth, or sometimes the deeper tissues which are not chaired, that also can be preserved for the DNA profile. I want to share my experience regarding the fetus. When we were receiving the fetus for the autopsy purpose, then the sample of blood on gauze piece was preserved for DNA profiling. But at some of the centers, I have observed that the whole fetus was sent for the DNA profiling. So that is another kind of practice. In sexual assault cases, where the history was there of sexual assault during the autopsy, we preserve swabs from the vagina and rectum. So according to the history, alleged history, we preserve the samples from the swabs and slides. I've also observed that in living cases, especially in sexual assault cases, buccal swabs and smear slides from the survivors and accused. Some of the centers, this kind of practice is there for the DNA profiling. And at some centers, blood in irrigated tube is the choice of sample to send for the DNA profiling. So these are the different types of samples which were preserved. I have observed or I have noticed that at some of the centers, different type of samples has been preserved. So here my suggestion is that there must be some guidelines and SOPs like we are having in Punjab, Haryana, Kerala. If SOPs and guidelines are there, then that can be followed uniformly in a particular state. But in the absence of that, different kind of samples at different centers will be preserved. And here we can see that CDFD Hyderabad is having the guidelines, and they are mentioned on the website. And they have clearly mentioned that what kind of samples they accept and how it should be preserved. In similar manner, I've also observed that John Hopkins DNA Diagnostic Laboratory, they have also mentioned very clearly that what kind of samples they will accept, how it should be preserved, how it should be transported. Even they have all clearly given. So such kind of the guidelines should be sent from the forensic science laboratory, and that can be helpful for preserving and following the all guidelines, and the better results can be taken out. So I have mentioned all these guidelines which are shared on the website of the this John Hopkins Institute and CDFD Hyderabad. Even B.N. Mathur sir has also shared the slide to me that they have also given the guidelines to the various centers in Rajasthan that what kind of samples can be preserved. So thank you all about that.
Thank you, sir. I now invite Dr. Jyoti Barua. I now invite Dr. Jyoti Barua, our next speaker. She is presently working as Associate Professor in Department of Forensic Medicine and Toxicology at AIIMS Bathinda. Dr. Jyoti has done her graduation, post-graduation and senior residency from Maulana Azad Medical College, New Delhi, and has experience of about 9 years of doing medical legal work. Prior to joining this institute, she worked in Uttar Pradesh, Chandigarh and Dehradun in reputed colleges. She has published more than 20 research publications of national and international repute and has delivered various guest lectures. Today, Dr. Jyoti will share her experience in sample collection practices. Dr. Jyoti.
Dr. Akhilesh, if you permit me, then I would like to speak something with Professor Mathur.
Sir, yes, sir, please.
Because I have to go for some other meeting. Professor Mathur, since you are a very senior person and you are an opinion maker, and
Yes,
in this group I am seeing some police officer is also attending this meeting. Do you think that the police doctor is not important for this developing this science?
Yes, sir.
This doctor means I have the experience like as a medical doctor. We are attached to the hospital. I was attached to the BHU, and the practice was there. Everybody of the whole district was coming to the BHU, and 2 medical officers from the state government were being appointed to do the postmortem. In fact, the specialist of eye surgery was doing the postmortem because he was never exposed to do the postmortem. I don't know how much he has learned during these undergraduate courses. So I would like to your opinion. I think you should, if you are of opinion, then you forward the opinion that yes, there is a need of police doctor because all these practices, like preserving the crime scene, getting good samples, needs education to the down level. The police goes at the side the first time, or the people, the doctor don't go there first. They get the sample.
Yes. Sir, your question is very valid.
I was able to explain my question or not?
Yes. Your question is very valid. And in fact, we also tried to depute or rather recruit some doctors for FSL for giving medical legal guidance to the forensic experts and to the other people also. But doctor's profession, there are 2 different departments. One is medical department and other is home department, under which the forensic lab comes up. Then the doctor, if he is posted at one place in a forensic lab and the crime is around the entire state, you have to visit entire state. So one doctor, how can he visit regularly, repeatedly, every day and to attend so many bodies? And then FSL don't have any postmortem rooms also, sir. So for postmortem rooms, we require highly specifications also, sir. So since we don't have it, but the medical jurists are always available. And I agree to this point that the peoples in villages and in towns also, they don't have a autopsy experience. As you told that eye specialist is conducting a postmortem. That is why I wish you that we must emphasize for study of forensic medicines for a large number of medical students, and they should be trained both for medical jurist profession, medical exemption, and they must be associated with the forensic lab also. As I told you in the very beginning, there was a controversy in a double murder case in Jodhpur High Court, whether the cause of death is brain injury or it is potassium cyanide. So since I was having a coordination with Dr. Vyas and I was having a knowledge of it, so I immediately, when High Court demanded some literature on this, I gave a ring to Dr. P.C. Vyas and he sent me so many books. So recruitment of a medical doctor in forensic department or in police department will not suffice our function. Let train every doctor. That is why now you will be happy to know that even in law department also, they have introduced forensic for 5-year law graduates. In 5-year law graduation course, 2 semesters are specifically meant for forensic purposes, forensic studies. At present, I am teaching in Manipal University. It is a very, very highly prestigious university in the country. And you will surprised to know about the syllabus which these law students are reading. It is starting from initial morphology of the body, anatomy of the body, physiology of the body, basic fundamentals of anatomy, physiology of the body, then about the poisons also, and about the latest techniques also, that light detection, polygraphy, narco analysis, DNA profiling, brain mapping, all that. Despite of this fact, I don't understand the medical students are not on. So this should be introduced in your MBBS course. Forensic should be introduced specifically. This thing will be there. And
Yes, sir. Yes, sir. The government
There
has
should be a specialized police doctor available in all the district so that they can attend the crime scene and they can do good sampling. I am a
Yes, sir.
vivid reader of the Robin Cook books, and there the analysis of autopsy, like at molecular level, he describes the how he identified the culprit.
Yes, sir. Yes.
Yes, sir. The
I do
government
agree with this.
Even if you read those books, then you will know that we are not doing anything in India because basic post-mortem and basic sampling, if it is not correct, then definitely you can't build a case.
Yes.
That is why you will. Those who are joining this course, I think we should start like writing government to start the many forensic universities so that they can produce the good doctors to analyze the things and to do good analysis.
Sir, in the
Yes, sir.
very beginning of
Government are taking this into the consideration, and at some of the states like in Punjab, in UP, and Maharashtra, they have already in the process of recruiting the forensic experts at district level. So recently, the recruitment is under process. Especially in Punjab, so many seats are lying vacant, and they have already advertised that. Even in UP, for medical legal expert, that will be take care of the autopsies at the district level. So it is going on, sir, and government is taking into the consideration of the same, sir.
Yes, it is in very slow motion because I know personally the status of the forensic universities or Raksha Shakti universities in different states. So the progress is very, very slow.
Yes, it is, sir.
Sir, despite of this, you will be remembering that in my initial lecture, before I started lecture, I gave a vote of thanks to three dignitaries to whom we have invited. Sir AIIMS Vyas, he has established the first forensic university in Asia and probably in the world. He is doing hard work for developing best forensic scientists in every field or in individual fields also. Then Honorable Justice Nandini Vyas, she is taking care of judicial officers and all, and then Dr. Vijay Lakshmi, Manipal University HOD, she is taking care to groom best advocates. And similarly, I want to bring in your knowledge that in Jaipur, there is one institute, Indian Institute of Health Management. They are in that institute. Doctors from the rural area of the country and abroad, they are regularly coming to read forensic, how to collect the samples, how to analyze the samples, how to interpret the forensic reports and the medical use reports. So, and I am a regular visiting professor there also. So your question is very valid, but please remember, we have to give more and more emphasis on your trainings at hand, practical trainings and that. Because now there was a question that how to preserve sternum. For bony, any bony bone, you don't require any preservative. The bone should be complete only. It should not be cracked, because in a cracked bone you get lot of infection, and you want to extract DNA, you will extract DNA. But the DNA of the microorganisms who has already infected the bone or the tissue, they will also multiply in PCR. So your original DNA will have less amount, and the DNA of the other bacterial flora will be more high. So your stand is very nice. We can recruit some doctors also, and it should be recruited at not only for the major forensic labs. It should be recruited at every regional, at every regional level also. So then we will have a lot of doctors for conducting postmortems and for interpreting forensic and medical reports also. You see that in the initial stage, I explained one the cause of death, whether the cause of death is brain injury or potassium cyanide. And ultimately, I explained the modus operandi of the crime also. The initial KCN, potassium cyanide, was given at the initial stage. But the fatal period of potassium cyanide is 3 to 4 minutes. So it cannot cause any fatal action during these 3-4 minutes. And during this period, it has circulated 100 times, 300 times, 400 times. And then the blow was given to her. So it will be a coordination if your project gets successful, and recruitment of the forensic scientists and the medical jurists should go side by side. It should go side by side, and their training should be gone side by side. It is not easy that you recruit a one person in forensic lab or in a medical jurist. He will become an expert at it. My expertise is my complete dedication. I used to do work from my own hands, and my teachers are very high. So we require extensive trainings also. So by side recruitment, put them on trainings also, both regular book training.
A day before, I read in a newspaper that they created the finger impression on a rubber stamp, and they used it. I think this is a society to find out the solution of this, because after some times even this finger impression will not be valid.
Yes, sir, it is available on Amazon.
Yeah, a day before, I read in the newspaper that they created a stamp of your finger impression, and they used that finger impression for taking the money on the online source.
Sir, this is implementation or transferring of the fingerprints. The nature has given us two characteristics for 100%: fingerprint and DNA. But please remember, fingerprint is not genetically innate. It can be translated very easily with the help of a sellotape. But when you will see it, you read the poroscopy. We have pores in between the ridges of the fingerprints. If the person is alive, the pores of the sweat glands are open. The sweating is coming out from the pores between the ridges of that. And the pores are also specific for person. The shape of the pores, it has many shapes, and the dimension of the pore, the number of the pores per centimeter in fingerprint ridges, they can be helpful also. And after that, the pores gets closed because the sweat glands are not working. So
thank you, sir.
I have a keen interest in this area. After listening Professor Lalji Singh, and I was meeting Mr. Gandhi, the one who is running I think, organization Truth in Hyderabad. He is a ex-police officer. So,
Truth Lab. Truth Lab. It is Truth.
Truth Lab. Yes, yes.
Truth Lab. Yes.
So many times I had discussed many things with him.
Yes, yes.
Even the ethical issue in the lie detection.
Sir, discussions are always fruitful.
Yes.
But why the court has not admitted lie detection, narco analysis, and other brain mapping, etc.? It is because of the reason that these all are controlled by the involuntary activities of the body, and which can be sometimes checked by the medicines also. Sir. If the person is under sedation, he cannot speak properly. So we do it, but court will not admit it. If we are doing lie detection and we are doing narco analysis, the person has already consumed a sedative, so he will go on sedation. He will not speak anything. So, sir, we will be required to sit with you for discussions and all that. Make a good plan, and you know that a DNA bill is also under consideration in Lok Sabha and Rajya Sabha and in the Parliament also. So, but your action to recruit more and more scientific persons, send them for specialized trainings. Since I was the only person in a forensic lab in 1972 to deal with a lot of crimes, and I have my own specifications in human body. So I could explain the crimes against human body very well. Fingerprints can be, but when you will lift the fingerprint, observe it under the microscope, you will find you have another field of investigation, that is poroscopy, study of the pores between the ridges of the fingerprints. So, in a
permit me to leave, but I have to go for some other things.
Thank you very much, sir, for your cooperation and inviting me. And we must sit together with all these eminent guests to whom you invite. They will give you fruitful guidance and give a thrust to your project for recruitment, for training, and all that. We can have a MOU with so many universities in our country, medical and legal and law universities, etc.
Thank you.
So we will be meeting very soon, sir. Very soon. Thank you very much, sir, for calling me.
Thank you, sir.
Thank you, sir.
Sir,
Thank you, sir,
sorry for the interruption.
So thank you, sir, for your suggestions. Your suggestions are well taken off, and we will come out with some guidelines, or we should say some kind of request to the government so that they can implement these forensic people, the forensic medicine people, into various cadres and categories. So now we are inviting our next guest speaker, and she is waiting patiently. She is waiting patiently, Dr. Vimukti Chauhan, and okay. Before that, I would like to invite Dr. Jyoti Barua. So she will conclude her talk. So Dr. Jyoti, please.
Thank you, Dr. Ajay, for your kind words. It is, as sir has already mentioned regarding the places where I have already worked, so I'll be sharing my experience pertaining to those areas, those places have the common practices that are being carried out for collection of DNA practices. Most of them are a repeat of what Dr. Ajay and Dr. Pathak has already said because of the universal guidelines by the Ministry of Health and Family Welfare. So most of them are repeat, but few of the current practices that I would like to mention, the eminent faculty from forensic as well as the forensic scientists from serology and biology division would be in a better position to comment regarding the amount, the type of samples that are being taken. A few of the samples I would like to say are merely taken out of the convenience of the person who is collecting the samples. So beginning with the various broad categories under which I would like to describe how or what the samples are being taken. Among the fresh bodies, as during post-mortem examination, as the body has been opened, so the sternum being easily available is usually being sent. Whole of the sternum is being sent after removing or cleaning the soft tissues. As Pathak has specified, a glass container was being used, but what I have experienced commonly, a plastic container is easily available. So it is usually being sent in a plastic container after cleaning the soft tissues, and no preservative is added after cleaning the entire sternum, the bone. No preservative is being added. Tooth in fresh body is rather becomes difficult to extract. So as I had said, because of the ease of availability, sternum, since being removed during autopsy, is being usually sent. Tooth, if extracted or removed, the molars are the preferred source of sample. The tooth, after being extracted, is usually wrapped in cotton and again placed in a plastic container. Cotton because of to avoid any friction or by coming in contact with the plastic again and again when it is being transferred. So it is being wrapped in cotton and then placed in a plastic container. Again, no preservative is being used for its transport to the FSL. In decomposed body, tooth are easily extractable, so there's no issue as it was present in case of fresh bodies. So decomposition easily falls off, so not much of expertise is required for extraction of the tooth. It is being extracted by a tooth extractor. Again, the same method is being used by wrapping it in cotton and placing it in a plastic container. Sternum, if tooth is not available, usually a single sample is being sent. So if tooth is being sent, so sternum is not sent in those cases. Muscle tissues are also being sometimes sent, about 100 grams of the muscle tissue, preserved in saturated salt solution and sent in the plastic container as the other routine viscera is being sent. So it is again preserved in saturated salt solution and then sent for analysis. In case of dismembered body, depending upon the body part which is being sent for brought for examination, if the skull is being brought for examination, then the tooth would be present. So tooth would be the preferred sample. In case the head is not present, then depending upon the tissue, any muscle tissue is being sent in a similar manner as with the case of decomposed or a fresh body. In case of skeleton remains, again, depending upon the type of the sample or the portion of the bone which has been brought, if the skull has been brought, tooth would again remain the best sample that is being sent. If in other cases, if it is not present, then depending upon it, the portion of the bone tissue, smaller bone is usually preferred because larger bone is difficult to transport. So a smaller bone, if it's present, then it is being sent, wrapped in cotton and again placed in the plastic jar without any preservative. In case of charred bodies, whatever portion of the tissue or whatever portion of the bone can be recovered is collected carefully and then placed in a plastic jar, again without any preservative. Fetus. Now the cases of fetus are very interesting, being smaller in size. Usually it is also possible that the entire fetus can be sent for examination. In my experience, what I have experienced is I have also been a member of the board cases in which the entire fetus was being sent for examination of DNA. There was orders by the court to preserve the sample for preservation of paternity testing. That is, there were cases of sexual assault in which a board was being formed and termination of pregnancy was being done. So we were supposed to preserve the sample of the fetus and of the aborted fetus or the fetus which has been terminated after the court order, and the entire fetus was being sent. Now, what issue arises in this is that transportation. Now, this will be valuable inputs will be given by the eminent people from the eminent forensic scientists from the serology and biology division. They will be in a better position. They will comment about how the temperature requirement should be maintained during the transport, because during my experience, I was informed by the people from CFSL that a temperature is to be maintained. So I had to make a lot of efforts in providing the container for the transportation, which was not available by the law enforcement authority. So I had to arrange for all those procedures. Vaccine container was being asked, and I had to preserve ice in the vaccine container, and I had to send the entire sample. Such. Now, there is no problem if the FSL or the CFSL is close to the place. In my experience, what happened is the place, the state in which I had to set the sample, their DNA machine was not working. So I had to transport it to another state. Of course, there was a coordination between those people at that particular state, but there are certain technical problems which arise because of transportation. So if during the panel discussion, if it is suggested any alternative method has been suggested, if only a small portion can be sent, then it will definitely be of valuable help. Another sample which is preserved in case of fetus is the long bone, or if it is a freshly aborted fetus, then sometimes dried blood on gauze is also being sent. In the living, usual scenario, the cases of sexual assault and of maternity and paternity issues are being addressed. Sexual assault, I will not go into much detail, as Dr. Ajay had already exhaustively mentioned regarding the guidelines related to guidelines by the Ministry of Health and Family Welfare. So similarly, in lines with that guidelines, the samples are being taken, and particular care is being taken to air dry the sample, as had mentioned by sir. The liquid blood sample is being taken in case of survivor as opposed to the accused, which I'll talk about later. Because we are already drawing blood for other tests, that is for the investigations or for blood grouping. So similarly, we need not again take another sample. Rather, similarly, about 2 to 5 ml of venous blood is being sent in a PDTA vial. Moving to the accused, dried blood on gauze is usually the preferred sample because of the longevity of the sample, which can be placed. That is, for years it can be. Sample can be analyzed. FTA cards, again, depending upon the availability of the subjective availability of the cards that Dr. Pathak has already mentioned regarding the FTA cards. So subject to the availability, in all circumstances, these cards are not available. So if these cards are not available either with the department or with the police officer who are bringing the request for examination, so what we do is we withdraw some amount of blood and we smear the small piece of gauze with the blood sample, which is then air dried and then again placed in an envelope. No preservative is being used for sending the dried blood on gauze. In maternity and paternity issues also, again, The blood sample is the preferred choice, in which the sample is taken not only from the child but also the alleged parent in question, in order to establish the paternity issue. Thank you.
Thank you, Dr. Jyoti. So now we are moving towards our next session for BCME.
I now invite Dr. Vimukti Chauhan, Assistant Director and Scientist C at CSSL Chandigarh. Ma'am is a recipient of Union Home Minister Award for Outstanding Contribution in the field of forensic science, in recognition of her valuable contribution to forensic science services, excellent and innovative contribution as a project coordinator, and a keen researcher. Dr. Vimukti, for over more than 2 decades, has been a vital resource in helping in various projects of advances in proposed computerization of CFSL. She has published many research papers in the field of forensic science and has played a major role in the publication of documents for quality system as well. Her exceptional contribution in promoting and supporting quality system have been highlights of a vibrant role in forensic science. In addition to this, Dr. Vimukti Chauhan has been deeply committed to and involved in the field of toxicology and has reported thousands of cases, assisted in judicial inquiries pertaining to death of prisoners, and has led the team of crime scene investigators. Her creativity, enthusiasm, and love for chemistry made her a consistent performer throughout the career. She is a well-known and highly regarded as a creative problem solver who consistently demonstrates excellence by efficiency, productivity, and quality of her work. Today, ma'am will be speaking on the topic, collection of biological samples for forensic analysis. I request Dr. Vimukti to please share her screen.
Good afternoon, everyone. I am Dr. Vimukti Chauhan. Thank you for the kind introduction. I am really grateful to Dr. Pathak, Dr. Ajay and his team of the organizing committee for giving me this opportunity to speak in front of these wonderful delegates. Thank you, Dr. Saab. 2 days ago, when one of your colleagues asked me for my photo for the brochure, you really scared me. You know the reason? Because in one of the incident, one of the police station was communicated with my phone number and name for the official purpose, and the enthusiastic police officer just displayed my phone number and my name along with the most wanted criminals. And it is read as Inami Badmash, Dr. Vimukti Chauhan. I am very thankful. I'm very thankful that this time the results are quite appreciable and my name is appear along with very eminent scientists. Thank you, Dr. Ajay, for that. And thank you the police officers who have displayed my name as a Inami Badmash also. So let's just not waste the time of the delegates and dive right into the presentation.
Ma'am, please share your screen.
Yeah, I'm sharing it right now. I hope it is visualized to everyone now.
No, ma'am. Reshare, ma'am. You try. Please try to share it again.
Okay, I'll do it again.
यहाँ से भी तो कर सकते हैं मैडम की स्क्रीन है ना
Mahi sharing.
can be from our end.
We are trying to share it from our end, ma'am.
Is it okay now? Dr.
Yeah,
Ajay?
Yes, madam. Yes, madam.
it's
Yeah,
okay
it,
now.
yeah, it is visible now.
You
Okay.
can do it in full screen mode.
Yeah, I'll try. Now it's okay. Is it in full
Ma'am,
screen mode?
no, it is not in full screen, but still it will do. It will be fine, ma'am. You can go ahead, please.
Okay. Okay, so the topic of my today's presentation is sampling of forensic toxicological analysis, because without the proper samples, we cannot go for systematic toxicological analysis. There is no use of investing crores of rupees in buying and adopting the technologies until unless we are not good in taking the samples. So the reliability and accuracy of any toxicological result is usually determined by the nature and integrity of the specimens. Specifically in case of samples where results goes to judicial system. A specimen from living objects, here urine is one of the very important and best specimen for comprehensive drug and poison screening in urine, because the accumulation of drug poisons and metabolites in urine will often result in high concentration. Here I want to emphasize that a minimum 30 ml sample in post-mortem cases of urine and 8 to 10 ml in living subject case should be provided to the toxicological analyst. Sodium fluoride as a preservative should be encouraged. Another specimen which should be drawn from the living subjects as well as from post-mortem cases is blood sample. A minimum of 20 ml sample preserved in fluoride salts is recommended in post-mortem cases. Definitely it has to be preserved with the sodium or potassium fluoride salts. An additional 5 ml blood samples should also be collected containing a clot activator. In cases of, here are some vacuum blood collection tubes, vacuitainers, with their color code, which define that which color code used for with which additive. Plain tubes as a red, dark red as clot activator. In forensic science, most of the time ETTA tube and plain blood vials. ETTA tube and plain blood vials are handy and sent and are available in quantity of 1 to 6 ml or 5 to 10 ml. Hair samples is also one of the major samples which needs to be drawn from living subjects. Around 5 to 20 gram of hair should be collected from the vertex posterior on the back of the head by cutting as close as to the scalp as possible.
Excuse me, ma'am.
Yeah.
The slides are not moving, ma'am. It is still stuck on the first slide.
Just a moment, Dr. Sharing okay once. Even entire screen. Yes. Now is it visible?
No, ma'am. No, ma'am. We are sharing it from our side. We are trying to share it from our
just a moment, just a moment.
Yes, ma'am.
Ma'am, we have shared it from our end, and then now you can proceed.
Okay, so Dr. Jyoti, we should move to 4th, 5th of slide. Can we move it to that slide? Next. Yeah. Hair samples, I have already explained that 20 gram of hair samples should be collected from the posterior on the back of the head by cutting as close as to the scalp. It should be marked that which end is closest to the scalp. They should be kept in a plastic collection tube or paper envelope. Ambient temperature is suffice for their storage. Next, ma'am. Next slide, ma'am. Yeah. Postmortem specimens should be collected as quickly as possible. That is a thumb rule. All samples must be collected in separate container. For most specimens, disposable hard plastic or glass tubes are recommended. Dr. Jyoti has just told us that what type of containers has to be used. At the end of the presentation, I'll explain in detail about that. Samples should be stored at a maximum of 4 degrees centigrade when analyzed promptly after autopsy. In case of liquid specimens are to be frozen, it is recommended that at least 10 to 20% of head space in the specimen tube should be left vacant. Next, ma'am. Yeah. Blood in post-mortem cases, blood remains the single most important specimen to analyze. I already told 30 ml of central blood and 10 ml of peripheral blood is useful for qualitative and quantitative analysis. A preservative fluoride salt is mandatory unless suspicion of poisoning with fluoride or fluoride producing compound exists. Next. The advantage of urine is already explained. Now it's time to know that what is the disadvantage of urine occurs in instances in which death occurs very rapidly after exposure of a drug and poison. In such cases, while evaluating the urine report along with other exhibits should be taken care of. Sodium fluoride as a preservative is again encouraged in case of urine also. Next slide. Vitreous humor, not very popular one, but very useful one. The fact that vitreous humor resides in an anatomically isolated and protected area of body, coupled with its good stability as a biological fluid. Presently, it's not a popular sample in forensic field in India, but it is very useful. It should be preserved with sodium fluoride in 10 milligram per ml. Next. Gastric content, specifically in the cases of pesticide poisoning, which is common in India. Gastric content is quite useful. Gastric content along with 100 gram of stomach walls in a plastic container, as well as 30 centimeter of first part of a small intestine in separate container, preserved in saturated solution of common salt is very, very important in case of pesticide poisoning, as well as when the death is quick. Next. Bile, many drugs and poisons have been shown to concentrate in bile. When collected, all available bile should be preserved with sodium fluoride, 2% weight by volume. In order to avoid the fermentation of this specimen, it is necessary that it has to be stored at temperature at least 2 degrees centigrade. Next. Cerebrospinal fluid is again not the popular one, but very useful, should be taken care of. It is also likely to be contaminated, less likely to be contaminated or have bacterial infiltration. While there are only few published references about the drug interpretation, it is a useful specimen for screening of drugs. Collect all available for in a 10 ml plastic container, leaving head space. Next. Tissue samples collected in post-mortem investigation is usually a general practice all over India, and it is analyzed for a systematic toxicological analysis. All the tissues presently preserved in saturated solution of common salt. Next. Postmortem tissue samples, liver. Around 50 to 100 gram of tissue should be collected preferably from right lobe and preserved in saturated solution of common salt. The advantage with the liver is that most of the drugs and poison are metabolized in liver, and both the parent compound and its metabolite may be present in high concentration. So liver is a good sample, sand it in 50 to 100 gram quantity. Brain, since the brain is the primary site of action of many drugs, so it's a relevant sample, specifically in cases of lyophilic drugs, drugs of abuse, organochloro insecticides. 30 gram preserved in saturated solution of common salt will suffice. Next. Lungs. The apex of either lung is the best choice of specimen for STO. It is a good sample in case of high concentration of drugs and poisons, specifically those cases where inhalation and intravenous exposure has been encountered. Next. Yeah. Kidney. Kidney, most drugs and poison metabolites must pass through the kidney to be extracted in the urine. So in case of heavy metal poisons, kidney is a very useful sample. 25 gram sample of kidney is recommended for the submission of metal poisoning specifically. In few cases, when apparent injection sites are visible, subcutaneous fat and skeletal muscle tissues should be sent along with fat layer. A control sample from a comparable area from the suspected injection site should also be collected. Next. Hair and nails. These specimens allow for an assessment of exposure weeks to months prior to collection. The best collection site for hair is from the vertex posterior of the back of the head. Strands of hair should be cut as close as possible, kept in an envelope. Storage at ambient temperature is sufficient. That is the advantage of this sample. Whole nails should be removed from the fingers or toes and stored in a manner similar to hair samples. Next. Next. Yeah. Entomological specimen collected from human remains may be useful for STA. However, it should be noted here that the concentration of drugs and poison in these specimens is dependent on the tissue that the larva had fed upon and their developmental stage. Next. In many cases, the evidence found at the scene provides the drugs, peripheral, cups, bottles. They should also be collected and sent by the investigating officer for STA. Next. I am summarizing the whole presentation in the form of this table. Blood, heart, always, specifically in trauma cases. Urine, always for drugs and their metabolite identification. Bile, for complete toxicology testing. It is necessary that collect the bile prior to liver, the off gallbladder to reduce contamination for heavy metal poisoning and exposure to other poisons. Vitreous humor, I already told you, is not the popular one, but very useful one. Combined fluid from both the eyes in a single tube. Preservative, I already speak about. Gastric content. Tie off stomach to prevent contamination of other viscera. Note total volume if possible. Next. Here, the liver from deep right lobe should be preferred. Kidney for metal, ethylene glycol, lyophilic drugs, hydrophilic drugs identification, 25 gram to 50 gram sample will suffice. Spleen in carbon monoxide and cyanide poisoning, Very useful when blood not available in the fire deaths. Brain and small intestine is also a useful sample in infant drug deaths, and lungs for volatile poison. Here with drug history and metal poisons. Next. Yeah. Now I am talking about the bone of contention highlighted by Dr. Jyoti. Ma'am, I'm coming to the concern you have raised. This is a very severe condition faced by almost each and every forensic science laboratory, including Central Forensic Science Laboratory. In order to avoid putrefaction of viscera samples, they are taken in a clean, wide mouth containers properly. Nowadays, Pearl Pack PP containers are available, which is good to avoid breakage during transportation. Next. Here I am showing these type of containers should be used for samples, viscera samples, and this type of container we can use for carrying these viscera samples. The container may be in the yellow color, must be in yellow color. Nowadays we got these samples in corrugated boxes, which are not good enough, and a lot of mixing happen during the transportation. So avoid those things, those containers. Use a plastic HDB container to carry to these viscera containers. Thank you. Thank you, Evan.
Thank you.
Thank you, Dr. Vimukti. Thank you so much for specifying the exact sample and the amount that has to be collected for toxicological analysis. Now, moving on to the panel discussion, I now invite eminent speakers, Dr. V.N. Mathur, Dr. K. Thangraja, and ma'am, you also please stay us on the platform, Dr. Akhilesh Pathak, Dr. Ajay Kumar, myself, and Dr. Ratan Singh. In the panel, we have received a lot of questions during this session, which we'll be discussing at this platform.
Dr. Thangraja, are you there, sir?
Yes, I am here.
Okay. So again, we welcome, once again, we welcome our speakers, Dr. K. Thangraja, Dr. V.N. Mathur sir, and Dr. Vimukti Chauhan. So all are here for sharing their knowledge on the questions asked by the participants. So, sir, I am taking the questions asked in chat box one by one. Dr. Rajendra Koleri from Bikaner, he is having one query. Is DNA profiling is possible in blood collected from decomposed body? So, if we are collecting blood sample from decomposed body, in where the chances are of hemolysis of the blood is there, so can we get DNA profiling from that one?
Yeah, so even if the DNA is very badly degraded, so there are markers which can amplify very small fragment. STRs also can amplify less than 100 base pair. We can use mitochondrial DNA, which even you can amplify 50 base or 70 base pair, and we can reconstruct the mitochondrial sequence. That's still possible. When I presented a bone sample of thousands of years old, this is nothing.
Yes, sir. Thank you, sir. So it is my also experience. It is also my experience in Rajkot that in some of the cases we sent the blood from the decomposed body and we got the positive results. Thank you, sir. So another query of the Dr. Rajan Kuleri is there. During MTP, there is little product of conception found in early pregnancy and preserved in saline. These product of conception dissolve in preservation after some duration. Is DNA profiling possible in this situation? So what he wants to know that, sir, we are getting the autopsy cases where the product of conceptions are there in uterus. and that product of conception is taken out from the uterus as a sample and preserved in saline and sent to FSL. So can we get a DNA from that one?
Yeah, if it is a saline, absolutely fine. Yeah, there should not be any issue.
Okay, sir. Thank you, sir.
Sir, would you suggest the preferred method of preservation in such cases?
No, usually is a saline. If you want to keep it for a little more days, you can put it in the 70% alcohol or dry sample in the freezer, right? So these are the one.
So what would be the cutoff limit for example, if you want to preserve in alcohol? Cutoff limit 24 hours, 72 hours. How much duration can it be
No,
preserved?
for a few days is okay. If you put in 70% alcohol, it can remain there for a few days. And technically speaking, even if it's there for several days, several months, no problem. You can able to do. But to critically analyze, get high quality DNA for the older method like southern based DNA fingerprinting, then you have to preserve the sample in very, very crucial way. Either put that in the freezer, minus 70 degree, or in the liquid nitrogen. So these are some of the highly preserved high quality method. But for STR based methods, you don't need such a high quality preservation. Even sample left in the room temperature, one can able to isolate DNA and do the analysis.
Sir, these would be required for transportation. For example, as I had mentioned in my during my experience, there were two durations. That is one for preservation and the next for transportation. For example, the police officer was likely to come after 5 hours to collect the samples. For that time, I had to preserve it at minus 70 degree in a freezer, deep freezer. And for transportation, for example, I had to arrange for some of the vacuum tunnels. For that, what temperature is required and for how long can we maintain that chain of temperature?
If you already frozen the sample, and you can pack that in the dry ice in a thermocol box. Dry ice can stay overnight depending upon how many kg you are keeping there. So that's one of the best way of transportation. Or if the sample is anyway outside for several days, one more day is not a big issue.
Okay. Thank you, sir. Thank you for mentioning this.
Thank you, sir. The next question is, it is asked by Akhila SD, that if a person had undergone bone marrow transplantation, whether any possibility to change entire DNA of his blood, is it there or not?
No, that
is the question.
When persons do the bone marrow transplantation, it's not that completely are going to remove the individual's bone marrow. So there will be always a mixed profile when you do it.
Obviously, that was the simple question. We move on to the next question, which is by Kirthana Chaudhary. If the offender is a monozygotic twin, how to report a case based on DNA sample? Is there any method to find out the identity of the monozygotic twin to confirm him or her as a culprit?
So one way effectively is if they are staying in two different environments, there's a possibility of epigenetic difference. Recently, which we demonstrated, the people who are living in two environments, but they are with the same ethnic background, particularly the Tibetan population. They also are in high altitude, and the same population, of course, they migrated 50 years back to low altitude in Karnataka. We looked at the whole genome, a whole epigenome, and we found there are some signature changed between these two. Also, if you do the whole genome and the whole epigenome, you may able to find out some difference between these two.
Thank you, sir.
Good afternoon, Mathur sir. One query is there from Dr. Mohammed Masood. Buccal swabs in dead bodies victims of sexual assault should also be preserved. So there is suggestion from the Dr. Mohammed Masood, and the query is: can hair without roots are useful for DNA analysis? So hair without roots, can we take out the DNA from that?
Yeah, the answer is sitting in front of you, Dr. Thangarajan. He has done a lot of work on mitochondrial DNA. So if you have a sample with the root, you will be having a sample of piece or sample of skin also. That will give the nucleated cell will give you the entire DNA profile. But if you have a cut sample, then you have to refer it to Dr. Thangarajan for mitochondrial DNA matching or mismatching. So no problem for that.
Okay, sir. Thank you, sir. So it can
Your
be
object
retrieved.
should be clear.
We have to send it to the proper center.
The proper center for this mitochondrial DNA of the hair will be Center for DNA Fingerprinting Diagnostic, CDFD.
But it's difficult without root.
Okay.
Yeah, it is. I know it is difficult without root, but you can't ask anybody to give the sample. The crime samples, whatever the sample is available with us, we have to deal that sample. Once we have to take sample from the body, we have choice of the sample for collection. But from the other places, crime scene, we don't have the choice. So what are samples available with us?
I hope Dr. Maqsood's query has been answered. Next question is by Govinda Balmuchu. In which medium to preserve sternum or bone? I think that has already been answered. This question has already been covered up in the lectures. We move on to the next question by Sukalpa Rathore. That is, what gives the preference to molars over other teeth? This is the query of a PG student. Why molars are to be preserved then?
It is very easy to answer. The molars are larger teeth. They have large amount of the pulp also. Samjhe? So, and it is very easy to cut the molar also. And because it's a large teeth, it is difficult to cut a canine or incisor. Incisor is very thin. But molars are very big teeth. So, it is very easy to cut them and to make, they give a lot of pulp to us for identification. It has a large amount of the blood also in the pulp.
Sir, in case an incisor is being sent, so what would be the percentage decrease? For example, to what extent can DNA be extracted as opposed to molar than incisor? If an incisor is sent for analysis and if a molar is sent for analysis. Because incisor being the controlled part, most of the medical officers, what they do is that they extract the teeth that is visible, because sometimes due to rigor mortis, the jaw is clenched and they just avoid dissecting the tissues and muscles to open the mouth and extract the molars. So what would be the percentage or the efficiency or the sensitivity of detection of DNA in those cases?
We have a limitation of the sensors. A rigor mortis has been developed, then the doctor has to open or has to cut the tissues of the jaws also to collect the sample, best sample. Our aim should be to collect the best samples. When we are having our body, you are done, you have the postmortem observations you have done, you have already written that because of the rigor mortis, the clinches of the jaws has already taken place, you can't open the mouth. But when the autopsy has been done and rigor mortis has been written, there is no issue. You can open the jaw with the help of some separator also, and you can collect it, no problem. But in the inside, if you compare the molar with the incisor, definitely you see your own mouth. Incisors are thin and sharp. Incisors are flat and sharp at the places. So the amount of the pulp cavity is very less in incisors as compared to the molar teeth. So once the rigor mortis are developed, no problem. No problem. After conducting postmortem report and observations, you can just distend with some separators and take the molar teeth. No problem. It can be very easy digged, especially from the mandible also, because mandible is a movable jaw. It can be very easy to take. No problem.
Okay.
Thank you, sir.
The next question goes on to Dr. Vimukti Chauhan is, why quantitative analysis is not done in toxicological specimens, specifically quantitative?
Whenever it is asked in case of drug samples, quantitative estimation can be done if the availability of the prescribed drug or the therapeutic drugs standards are available with the laboratories. So it is not very difficult to get it done. The only thing is interpretation is not much informative in these cases.
Right.
Thank you, ma'am.
I just want to point out one thing, that in cases of drugs or narcotics, unless until you don't precise the amount of the narcotic substance in the viscera with percentage, I think that the case can be charge sheeted. It cannot be filed unless until that viscera shows the higher amount as compared to the benchmark amount. So in 1 kilo of the atta, you can have only 5 milligram any narcotic. It will show the presence, but it will not be offensive for filing an FIR in the court, honorable court. So along with the quantitation is the most essential part of it. And
So
if somebody requires training on this quantitation, he can be sent to the forensic lab Gandhinagar, Gujarat for training purposes.
So we have to specifically mention that we want a qualitative as well as quantitative analysis.
Quantitative analysis. It is necessary that you have to mention it, that qualitative and quantitative is required, and the sufficient quantity of sample is also provided with that. Normally we used to get hardly 1 ml of sample, and the questionnaire states whole screening of toxicological analysis.
That is the aim and object of such conferences. You guide the medical jurists how much you require, what are the difficulties you are facing for analysis and all that. You got a sample, but if you are not able to analyze it qualitative and quantitative, what is the purpose of collection of the sample and forwarding it to forensic lab? So these are the aim and object of such conferences. You guide the medical jurists that at least you do this thing.
Yeah, SOPs and guidelines. SOPs and guidelines has already been formulated and circulated to almost all the hospitals in that respect.
And the doctors should follow those SOPs also. Anyway, next question.
This is for Dr. Thangaraja. Is using alternate DNA marker more reliable over STR markers?
So one has to think about the application. The STRs are the one which is very highly polymorphic. If you wanted to find out the difference between individuals in the same family, because father, mother, and the child, all three can be differentiated by using the STR. So very highly polymorphic. But you cannot use mitochondrial DNA. Mother's, son, daughter's mitochondrial DNA sequence will be identical. Same way, fathers and sons or grandsons' Y chromosome markers will be identical. If you want to find out people from different ethnic group, then STR you cannot use, because that even after a few generations, STRs cannot be used to trace whether they are from the same ethnic background. So then one has to use evolutionarily stable markers like Y chromosome markers. One can able to construct the haplogroup by using set of Y chromosome markers, set of mitochondrial DNA markers that will tell whether these two individuals are from same common ancestry or not. So all depending upon what question you are asking.
Thank you, sir. The next question is, can RT-PCR of COVID testing used for DNA analysis? The person wants to ask that RT-PCR machine, can it be used for DNA analysis?
Yeah, it can be used. Yes, can be used, but there are different set of markers can be used. For example, I wanted to look at the SNPs, whether in a given position there is a C or T, right? So in that case, you have to design primer called Tachman probe, which can be used to differentiate. So in that case, you have to design one more primer, just ending one base before the actual SNP. You are designing primer with the 20 base pair for one SNP, 22 base for another SNP. Use dideoxynucleotide. There's a specific protocol. So that way you can use 20, 30 different SNP markers, which can give you the profile, which can able to use it. But it's not necessary for forensic application, because that machines are quite expensive. So the best method is STR using the normal PCR.
Thank you, sir. So, sir, we are having some queries and we have prepared some queries that may elaborate again the things and clear the issues. So first query was there that what you suggest that what is the best sample to be collected for DNA profiling in living. So if any person is coming like accused of rape or survivor of rape is there and we want to collect a sample for DNA profiling, then what you suggest that is the best sample?
For DNA fingerprinting, there's
Yes,
no special preference as long as the individual is willing to give blood, draw the blood, or if he's not ready to give the blood, then ask him to give saliva, just spit that in a container which consists of buffer which can lyse the cell, or if he's not even willing to do like few air samples, so anywhere you can isolate DNA. So there's no preference, all depending upon the willingness of the individual.
So the yield remains the same in all the cases?
Yes, any tissue you take, the profile will be identical.
Sir, next would be decomposed body. Decomposed body, what would be the
decomposed body? If the tissue is very badly decomposed, then take the bone marrow, and if not bone marrow also not there, then bone itself can be used, or tooth pulp. So this can withstand for a very long time. So among the bone, if it is a several thousands of year old, then petrous bone is very good for DNA source.
Okay, sir, if we have a large portion of the bone, then how much amount of the bone should be preserved? For example, if we are getting femur, pelvis, the entire bone cannot be transported. So we
the entire bone is not necessary. If the bone is of only few months or few year old, then you can easily isolate DNA. You don't need small piece of bone is sufficient. And if it is thousands of year old, one has to use different method for isolating DNA. So therefore, we need little more bone sample. So there is no standard protocol for the bone sample, all depending upon how old the bone sample is.
Okay, sir. You suggest that a small portion can be broken and then sent in a container. That will suffice.
Yeah, that would be sufficient.
Okay, thank you, sir.
One question is what type of samples you accept and what you do not accept for DNA profiling. Whether you have refused some samples citing inadequacy of the amount or that particular sample is not worth carrying out the DNA analysis.
No, we generally accept all the samples because it's always better to try rather than refusing it. So we never know, we may not see few cells on any material where we feel that the person who is bringing sample, there are few 1 drop of blood or even less than that. There's a air with or without root. We may not be able to differentiate whether root is there or not, but it's always better to try.
Okay, sir.
Sir, any particular contaminant which is likely to interfere?
No, I don't think anything will interfere unless otherwise there is a contamination of human blood, because the marker which you use, they're all human specific. So even there's a lot more contamination with any animal or bacteria, doesn't matter. But you are using the primer specific to human. So unless otherwise there is a human DNA, it will not give you any result.
Sir, even in charred body that we had mentioned, the charred portion of the tissue can also be sent?
If it is totally charred, it's very difficult. Even if the charred one, interior, one can able to dissect out and see inner portion if something is left, the actual tissue without totally burnt. So that can be used. Ultimately, what you need is just 1 nanogram of DNA for generating the profile. So few cells are sufficient to generate the profile. So that's the advantage of the DNA-based technology.
Sometimes it happens, sir, that the IA requests for DNA sampling on a very later stage, where in cases we have already sent the viscera for chemical analysis. So can the DNA again be extracted from those samples which we have already sent to FSL?
Generally, we send these viscera in saturated solution of common salt. So a viscera which is sent to you in saturated solution of common salt, can it be used for DNA extraction also?
Yeah, it can be tried after washing several times. More salt, even if you isolate DNA, it will inhibit the PCR reaction. So when there is an enzyme polymerase, will not work with too much of salt. So one needs to wash thoroughly to remove all the salt, then isolate DNA and use it. Even the paraffin embedded, where it has undergone for dehydration, fixation, everything, but still we could able to get the DNA. So one has to try with different methods until you succeed.
Okay, sir. Thank you, sir. Another query is, sir, from the Supriya Kole. Is YSTR electropherogram analysis at all markers it will show one allele, but at some loci it will be 2 in number. Why?
So that means there are particularly that I think DSY 389. So there the same primer amplified two different regions on the same locus. So one amplicon is smaller, other one is little bigger. So when you use that particular marker, it will give naturally two alleles because it presents that region in two different places on the same Y chromosome.
Thank you, sir. The house is open for questions
or any inputs from our eminent delegates.
I think all the questions have been answered. All the queries have been answered. Still, if somebody wants to ask the question to our eminent speakers, they can directly ask by unmuting themselves.
My question, I am Dr. Sharma from Shimla. What is touch DNA technique?
So Dr. Aditya Sharma from IGMC Shimla is professor and head there, and he wants to know that
what is stretch DNA technique?
What is stretch DNA technique? So
touch,
is there
touch DNA.
kind of technique, sir?
Sorry?
Touch DNA. TOUCH.
Touch DNA.
Touch DNA. Touch DNA.
Okay, sir.
Touch DNA.
Touch
No,
DNA.
no. I can answer it. You
Yes,
say,
sir.
it is a question, and people are selling some DNA sequencer that they are able to analyze touch DNA. But if I touch this glass, you can't analyze by DNA on this glass. Touch DNA means you are wearing several things. Say, for example, I am wearing this ring for a longer time in my finger. So this cavity of this ring, and I am wearing something locket and some thread on around my neck for a longer time. I don't remove them for washing and other things and all that. So many things are kept on our body. They are in constant touch with our body, and they get absorbed our peripheral cells during sweating or during washing and other things, because the cells from the body are regularly discarding. It is a regular process of removing the cells from the body. It is a natural process. So those cells, they get entangled in these threads or in these ornaments, which are in constant touch with our body. If you will remove it, you will get lot of DNA in this on the thread or the ornament which you are wearing. You might also be wearing some thread, black thread, or some golden chain, or some other chain, which is in constant touch your body. This constant touch in your body, it grabs lots of thousands and millions of the cells, remain entangled, and gets dried on those ornaments. So you can extract it from that. It is a wrong misconception for selling equipment that we are able to analyze the touch DNA. Some agency from the America and based Delhi-based office also, American-based office and Delhi-based office, they came to know, and they gave me that we are able to analyze the touch DNA. When I asked the insert, I am touching this glass, I am touching this pen, can you analyze my DNA? They don't know it. DNA, for DNA requirement, you require a nucleated cell, and you are wearings on the body which are in constant touch with your body. They get entangled. Millions of million cells on your thread which get dead, which you don't know. Even you wash them, you will get them. So that is the touch DNA. And yeah, that is a misconception. People are selling it for this. And Chandigarh laboratory, CFSL Chandigarh laboratory, they buy a next generation sequencer. So I told Askajan that tell me, ask him to isolate my DNA. I'm coming and I'm touching my this ball pen or touching everything on the table. So touch DNA means the ornaments or the threads or the rings you are wearing and which are in constant touch your body. Thousands and thousands and millions of the cells, they get entangled in those threads, and this thread which is in constant touch of your body, when it is analyzed, it can give your DNA profile. So this is a misconception of the examination of touch DNA.
Thank you, sir. Thank you.
Thank you, sir. Thank you, sir.
One more question is there.
Doctor, yes.
Is it possible to extract high quality non-destructive DNA from formalin or formaldehyde fixed human tissue? I think Dr. K. Thandraj has already answered this query that it can be, it is just we can try it.
Yeah, high quality we cannot isolate because high quality means without break. But when the process happens, DNA would be highly degraded.
Okay, sir. Thank you, sir. And another query is there that in advanced decomposed cases where organs are decomposed and lost, but partial soft tissues attachment present over the bone. So we are getting sometimes we are getting the skeleton where the bones are there, but simultaneously the soft tissue is also attached. So in that case, what we should send, or what will be the preferred sample for DNA, whether soft tissue or bone?
Yeah, the easy procedure for isolating DNA is soft tissue. If the soft tissue is good, then no problem. If the soft tissue is highly decomposed, very badly decomposed, then bone samples are the best.
To safer side, it is better to preserve the bone.
Yeah.
And okay, sir. Thank you. And Dr. Bimukti, in same case, when we are getting the skeleton with attached soft tissue, what sample should we send for the chemical analysis?
The soft tissue will suffice, and we can analyze with the help of those tissues.
Okay, ma'am. Anything else? Anything else from the audience?
I want to give some suggestion during this panel discussion. I came to know, especially with Jyoti Barwa and Vimukti, that they are forwarding the samples in plastic containers.
Sir, those are PET containers. They are generally called as plastic containers, but preferably they should be PET containers or HDPE containers. But in a common language, they call it plastic containers.
Yes, I agree with you. But do you name it that during transportation of the sample in a plastic container, anything can be put in the container from external media by meaning of syringe also. You can take any material, any poisonous material, and you can inject in a plastic container. But you can't inject in a polythene in a glass container. So be cautious. Never use plastic container. It is choice is up to you. It is choice is up to you. There are always possibility that anything can be injected in a plastic container through syringe, method of syringe. You are sending a viscera sample in a plastic container. I took it from one place or other. I put some poison in it. That's why no poison was originally present in the visa. Then I pushed it in the so it is easy to anything to be entered in a plastic container. Therefore, it is specifically always mentioned that the container should be a glass container which should be properly sterilized. This is my suggestion to you.
Thank you for your suggestion, sir. One more
the other
query.
thing, I want to finish my thing. The other thing that you have mentioned of collection of the blood on cotton gauze, which are available in the hospitals also. Have you ever seen the structure of a cotton thread? Why cotton is lighter? Why it is lighter? Because it has the structure of the cotton thread is identical to the structure of the DNA. It has large number of air vacuoles. So you think that you have taken a lot of sample amount of the sample on the cotton gauze, but the air spaces are not filled with that. It can absorb everything, but the amount of the sample required is very, very less. That's what we are seeking. So it is better that you should have a cotton cloth without any starch. So the cotton cloth which don't have any starch, it should be properly washed, and then you can take the sample. So no air bubbles will be left in that completely pressed cotton cloth. Otherwise, if you take blood on the cotton gauze or on the cotton, a lot of sample is wasted, because the cotton thread has the vacuums, has the cavities, just like DNA structure. So that is why it is vital. So always guide them to take the blood sample on a fresh piece of cotton cloth. Never use the gauze. It appears that you have taken more amount. So this is my suggestion to you, and rest. And if you get the bite marks on some body, it is to the doctors also. Some bite, you get a very deep bite marks in cases of rapes and others also. If you have some doctor orthodontist with you, you can lift that dental bite on just like you make a denture with the help of a powder that is available. It is known as lignite powder, available to all dentists. Just put it in water, make a steady solution, pour it on the bite mark, and leave it for 1-2 minutes, then remove it. It will give you the bite impression present on the cheek and all that. So thank you very much.
Sir.
Any more to say?
Sir, Dr. K. Thangarajan, sir, one query is there. Best sample collection for DNA for identification in case of completely charred remnant bone tissue after cremation. Sir, I have received one autopsy case, such type of autopsy case, when I was in Rajkot, that there was a history that one person was murdered and the cremation was done. And after getting the information, the police just collected whatever the ash and the bones after cremation were available, and they brought that to me. I sent it the remnant pieces of the bones for the DNA profile, but unfortunately I did not receive the positive report. What do you suggest, sir? Can we what we can get?
That requires specialized techniques for isolating DNA from the bone.
Yes, sir.
So and also depending upon the quality of the bone, for example, the bone is of 1,000-year-old, for example, or
Yes,
hundreds of year old. So that procedure is different than the fresh bone sample. What you are seeing is that somebody murdered, killed, and the body was charred. So that's a very fresh. So that will give you DNA with a very simple method. For isolating DNA from the old bone, we have to adopt different method, decalcification, and many more methods are there. So whatever the material, I mentioned that even the charred tissue or the bone still there should be some deep inner portion where the uncharred material may be available, so that can be used for DNA isolation.
So there is only possibility of that.
Yeah, possibility. Yeah. No, that's
yes,
what successfully we have isolated DNA from 1,000-year-old or thousands of year old bone samples.
Yes.
So this kind of samples are much easier to handle.
I hope that our panelists have answered all the queries. We have
Dr. Ajay,
no
I
more
have one more
questions
question.
left. Yeah, please go ahead, sir. Please go ahead, sir.
Yes, sir. Dr. Aditya is asking one more question. Yes, sir.
My question is for Dr. Thangaraja. Sir, just you have told that in charred body or partially charred body, you can extract the DNA. But whenever the body is burned, the temperature of the body rises maybe 1,000 degrees centigrade, 2,000 degrees centigrade. I think at that temperature, the DNA won't survive, whether you preserve the bone or whether you preserve the molar tooth or extra tooth. To forget about the soft tissue. So is it possible to extract the DNA in completely charred body, which has been heated to 1,000 or 2,000 degrees centigrade? Answer, please.
I'm telling not completely charred. So body must be charred, but still some portion of the bone remain as uncharred. If it is totally ash, it's very difficult. You cannot do that. If some portion, even after burning, some portion of the bone or even the teeth, which may be possible that it's better kept as it. So better to try in those samples rather than just say that it won't work.
Okay, sir. So there is a possibility of the unburnt tissue or live tissue in such cases. So if there is any live tissue is available, then it can be done.
Even you leave the tissue,
Yes,
even
sir,
the
definitely.
bone, even the bone. I'm sure that even the body was completely charred. There could be some portion of the bone. It could be of any bone, but still you can able to isolate DNA from that small portion of the bone.
So that means in those cases we should send all the remains of the sample that we have. We cannot pick and choose because we don't know whether it will be recovered or not. That means we have to send whatever sample we have received. All the entire samples have to be sent. In those cases,
you can judge yourself. Okay, so this is the portion which is charred. It's almost like a charcoal. So other portion, if you find something white color, is a bone is intact. So that portion you can send.
Yes, sir, my question is still exists. What is the threshold temperature for the DNA to survive?
Sorry?
Temperature at which the DNA survives in a human body.
So
80 degrees centigrade, 90 degree, 100 degree, what degree, sir?
So the 100 degree is the one which we use, not 100 degree, maybe 95, 96 degree routinely for PCR. Use 96 degree to make the DNA double strand into single strand.
So when the body temperature rises to 1,000 degrees centigrade or whatsoever it is,
correct.
So do we find the DNA, sir? Sorry,
so
sir.
No, what I'm saying is if you put the DNA directly in a burner, which is of 800 degree or 900 degree, it will not stay. But in a burning body, the bone is not going to expose to the entire 800 degree or 1,000 degree centigrade. So the bone, inner bone, may be of little less temperature, right? So there, if the bone is intact, if it is not charred fully, but you would expect the DNA may still exist there.
And that is also a rare possibility, sir is saying. If there is unburnt tissue is available, then only it can be extracted.
The unburnt tissue, the bone is still, you are able to see white color bone.
Yes, sir.
You can able to isolate DNA.
Yes, sir.
Thank you, sir.
If it is a completely charred black, then no hope in that.
Yes, if it is a ash, then we can't expect that DNA will be collected from. So thank you, sir. So now I would like to conclude this CME, and I would like to invite Dr. Ratan for giving a vote of thanks. Dr. Ratan, over to you.
Thank you, sir. As a good thing comes to an end, so has today's CME on a very pertinent topic, of DNA fingerprinting, past, present, and future. On behalf of the Department of Forensic Medicine and Toxicology, I, Dr. Ratan, take this opportunity to propose a vote of thanks to all those who have contributed to this academic activity organized in our institute. I would like to express my sincere gratitude and thanks to Honorable Chief Guest.
Muted.
Dr. Ratan, unmute yourself.
I would like to express my sincere gratitude and thanks to Honorable Chief Guest, our worthy Director, Dr. Dinesh Kumar Singh, who spared his valuable time and always been a source of inspiration. Unfortunately, due to persistent COVID pandemic, a physical academic gathering as planned could not be possible, but nevertheless, we managed to organize an online CME where esteemed resource persons enlighten us with their knowledge and experience. My heartfelt thanks to all our respected guest speakers, Dr. V.N. Mathusa, ex-director CFSL Rajasthan, Dr. K. Thangarajan, Director of Center for DNA Fingerprinting and Diagnostic, Hyderabad, Dr. Vimiti Chauhan, Assistant Director and Scientist, CFSL, Chandigarh, who took out time from the busy schedule and gave deep insight into the topic. And they also contributed in the panel discussion, which was very detailed and informative to us. I also extend my thanks to Dr. Akhilesh Pathak sir for his constant support and guidance, and Dr. Ajay Kumar for his valuable input in framing the CME, and Dr. Jyoti Barwa for her tireless contribution in the smooth functioning of the event. A word of thanks to the IT department of AIIMS Bathinda for helping us in managing the technical and audiovisual part. Last but not the least, I extend my sincere thanks to all the online delegates for participation and sharing of the knowledge. Now, in a short while, we will proceed for the plantation drive to make our AIIMS campus green. Thank you all. See you soon.
Before that, I just want to say thanks to all the eminent scientists present here, and a special thanks to the team of Dr. Pathak and Dr. Rajay, Dr. Jyoti and Dr. Ratan, who have endlessly doing work for this CME to get successful. Congratulations to all of you. It's a wonderful conference.
Thank you, ma'am. Before proceeding for the plantation drive, we would like to felicitate our guest speakers. Dr. Ratan will be presenting the mementos, and
yes, sir. It's online, and we will send it to you by the post.
We will be accepting on your behalf, sir. We will be sending to you physically by post.
It's a wonderful idea. It's a wonderful idea. Go ahead with it.
And thanks a lot, sir. All of to you, all of three to you. Thanks a lot from my team for accepting our invitation as a guest speaker and sharing your valuable time with us.
And thank you all the distinguished delegates who have spared their time for spared their time.
Bye to everybody. Dr. Mathu,
Pleasure is all ours. Pleasure is all ours. Thank you.
On your behalf.
Chauhan and others. Bye.
So we will be accepting mementos on your behalf. Dr.
Let's see.
Will be accepted by Dr. Akhilesh Pathak.
Sir, on behalf of Dr. B.N. Mathu, sir, I am accepting. It will be sent.
Thank you, Dr. Pathak. Thank you, Dr. Pathak, for accepting on my behalf. And
Dr. Ajay will be accepting the memento on behalf of Dr. K. Thangaraj.
Sir, I am accepting it on your behalf, sir.
Thank you.
Dr.
And I'll be accepting
and
the
Dr.
memento.
Jyoti will accept on behalf of Dr. Bimukti.
Thank you, Dr. Jyoti. This is truly touchless honor.
Thank you very much.
And my request is that such activities should go on constantly, continuously, and we should invite the resource people from every field, including medical, doctor, judiciary, OTC, and all this. So we should continue such activities, and thanks to all members of the organizing committee and the speaker. It is a good introduction also, and suggestions are has also been given by everyone. So thanks, Dr. Akhilesh. Be in touch with us. And,
Yes, sir. Thank you very much.
Thank you very
Thank
much.
you.
For
Thank you.
inviting me.
Thank you all the
very
delegates.
much. Congratulations for your hard work.
Thank you, ma'am.
ma'am.
Thank you very much.
Okay, Dr. Akhilesh, Dr. Pathak, Dr. Jyoti.
Thank
Thank
you,
you,
ma'am.
ma'am.